Hello guys, welcome to this session. And in this session, we are gonna start with virology. And virology is a very interesting part. You read about viruses, and trust me, it's not that boring like bacteriology. So we are gonna do virology through these notes. And since we have a lot to cover, I can just write and tell you everything. So we are gonna just look into all the important viruses which are important as per your PG entrances and in your Prof examinations. Let me tell you that your CBME curriculum currently focuses on a systemic approach, but here I am taking an organismic approach for this microbiology lecture. So for systemic videos, you can just follow your book and look at the viruses. The content is going to remain the same. Just if you have completed the systemic, if you have completed the microbiology, it is a bonus for you since you're going to get all the viruses in this sequence only. Okay. So let's start with introduction to virology. So virology, we know viruses we know are not living cells. They're obligate intracellular infectious particles. No nucleus cytoplasm are there. Size is in nanometers. Smallest virus is parvovirus. Okay, parvovirus. And parvovirus 19 is the common parvovirus to read medically significant one, and largest is the pox virus, right? Pox virus. So we'll look at the pox viruses, measles virus, rubella virus, and many other viruses. Will also revert parvovirus in the diseases caused by it, right? So viruses have either DNA or RNA. They don't have both. So never have both. They either have DNA or RNA. So you can have DNA viruses or RNA viruses. Some viruses have split genome, which you remember by just mnemonic Bora. Okay. So Bunya viridae, Orthomyxoviridae. Orthomyxoviridae has a medically significant only one virus, that is influenza virus, which I have to discuss in detail. Then the Rioviridae and Arinaviridae. Okay. So Rioviridae we have rotavirus. So rotavirus causes diarrhea, and we have to discuss rotavirus diarrhea in detail also, right? So adenovirus, how can remember? ADENA. Okay. So D and N and A, this letters are there. So it is a DNA virus, right? Okay. DNA virus. The organisms having a split genome has the capability of recombining, right? Reassortment happens, and reassortment creates novel viruses. Novel viruses in different strains. So must have heard about the influenza virus. There are many strains of influenza virus which are prevalent in our country in a periodic variation or a seasonal variation, right? This variation occurs due to a split genome concept only, right? When we come to influenza viruses, we will discuss how this is happening, right? Okay. All viruses have a single copy of genome haploid except retrovirus. Retrovirus has two copy of genomes. If you have read my previous read HIV or looked at my previous lecture which was on HIV, if you haven't, do watch it. It is a very good lecture combining your microbiology as well as the pharmacology, right? Okay. So retrovirus have a diploid genome, that is ssRNA. Positive strand ssRNA is present. Okay, and three enzymes are there: reverse transcriptase, integrase, and protease. Right, and we looked at drugs also which are inhibiting that. Okay, important examples are DNA viruses. So we can remember this is DNA viruses are happy ones. Okay, right. So these are happy viruses. So happy viruses like herpes virus. Okay, H2H are there, 1A, 3Ps are there, and Y. Y is just for making it sweeter. So happy herpes and hep DNA virus. Okay, hepa DNA virus or hep DNA virus for Twitter. That is hepatitis B virus. So herpes virus, hepa DNA virus. Okay, rest of the HIV viruses are hepatitis viruses are RNA viruses, right? Hepatitis viruses are RNA viruses except HBV. HBV is the hepatitis B virus. Then we have adenovirus, parvovirus, pox virus, papova virus. Okay, adeno, parvo, pox, papova. All three P's are there. Adenoviruses. And papilloma virus, we have to study. One is the polyoma virus, one is papilloma virus. So papilloma virus, we have to read HPV. Okay, we'll see how the risk, high risk HPV, what diseases it causes, low risk HPV, what diseases it causes, right? And polyoma virus. Polyoma virus, we have to remember one, that is the GC virus. GC virus is John Cunningham virus. Causes opportunistic infections in AIDS patients. Okay. So all the DNA viruses have dsDNA as genome. dsDNA, double stranded. Just a minute. Okay, so there was a call. So all viruses have dsDNA as genome, double-stranded DNA, but parvovirus have ssDNA. Okay, so parvovirus have a DNA which is line-like ssDNA. Okay, parvovirus B19 is the significant one that you have to read. So all viruses have genome and protein coat, which is known as capsid. Capsid is present in all the viruses, And two basic symmetries are there. One is the icosahedral, one is the helical symmetry. Icosahedral symmetry means just hexagonal type of symmetry, and helical, and there is a helical arrangement of the genetic material and all, right? So you can see icosahedral and helical symmetry, and this envelope is optional structure, so we can have enveloped viruses and non-enveloped viruses as well. So except this DNA viruses, all are RNA viruses, and in DNA viruses we have to remember which are enveloped and which are non-enveloped. Herpes and Hep DNA viruses, that hepatitis B, they are the type of rich viruses, right? So they have envelope is present. Adeno and parvo, envelope absent. Okay, pox virus is a good virus that so it will have the envelope, right? Pox virus. And papova virus, envelope absent again, right? So envelope is made up of phospholipid bilayer, and peplomers are glycoproteins. Peplomers are basically glycoproteins which help in attachment of virus to the host cell, right? So enveloped viruses are sensitive. Remember, enveloped viruses are sensitive to ether, alcohol, and bile salts. Okay, they are sensitive to ether, alcohol, and bile salts. The ether, alcohol, and bile salts tend to kill the enveloped viruses, right? And the non-enveloped viruses are not sensitive to this. Understood? Okay. So in GIT, bile salts are present. Okay, bile salts are present means which one will be more susceptible? Enveloped virus, right? So there is a enveloped viruses have loss of infectivity. So most of the viruses which infect the GIT are non-enveloped type, right? Rotavirus, Norwalk virus, calcivirus. Okay, this and picornavirus and this HAV and HEV also. Picornavirus and HEV are, we don't read much in GIT, but we have to read rotavirus, Norwalk virus, and calciviridae family, right? So enveloped viruses which cause diarrhea, coronavirus may cause you diarrhea. Okay, and CMV colitis. When we read about the cytomegalovirus, we'll read about CMV colitis here, right? And matrix protein is between the envelope and the nucleocapsid, okay, which will be interaction. Tegument is between envelope and nucleocapsid also, tegument, okay. And this tegument helps in the viral replication. So viral replication, let's come to how the viruses replicate. So first is the absorption or attachment to host cell receptor. Okay, this was cell tropism, that is a cell selection type. Okay, viruses attach to the specific receptors present on cell, like HIV, CD4 receptor as what was the co-receptor? CCR5, CCR2, and CXCR. Okay, CXCR was present on TH cells, and CCR5 was present on macrophages, right? EBV, Epstein-Barr virus, attached to CD21 receptor. Just remember. Adsorption occur by papillomas or capsid proteins. Okay. After the virus is attached, we have internalization. The virus will be internalized, right? So in naked viruses, nucleocapsid will enter the host cell, and in enveloped viruses, envelope will remain outside. Just the nucleocapsid will enter. Okay. So in naked viruses, we don't have envelope. Just the whole virus, you can say, enters the host cell. But in enveloped virus, the envelope leaves outside and the virus enters it, right? Then it is uncoated. Uncoated means capsid is broken down by host cellular enzymes. Okay, the nucleocapsid which is going there is basically uncoated. It's broken down. Okay, and the nucleic acid will lie freely. After that, biosynthesis occurs. The genome of the virus starts to multiply, and mRNA is formed. Right. Then we have the assembly of viruses. Okay, the daughter virion specifically, they assemble, and there is a release of the daughter viruses. Okay, how will the release occur? Lysis of infected cells and budding from host cell membrane in envelope viruses. So envelope viruses tend to take the outer coat of the host cell, but in naked viruses, if the naked viruses are infecting, then there is a lysis, cellular lysis, right? So you can see here in the diagrammatic format, the attachment is occurred influenza virus. Okay, influenza virus is here present. Okay, and influenza virus, you can know that if you see here, you can see here, the influenza virus is not coming under these categories. Okay, it is a polymyxoviridae class virus of polymyxoviridae family. So influenza virus is basically not is a RNA virus. Okay. Influenza virus, then it penetrates the cell, engulfs the virus by endocytosis. Okay, so attachment is there. Attachment is there. The internalization and penetration, you can say. Then there's uncoating, that's a breaking down, and the viral contents are released. And there's a biosynthesis. The viral RNA enters the nucleus and it replicates by viral RNA polymerase. And assembly means the daughter virions are assembled, and there is a release. Okay, there is a release means the virus takes the host cell Okay, which is an enveloped virus. Influenza virus is an enveloped virus, right? Okay. Next, we come to the how we can grow or cultivate viruses. Okay, so viruses are obligate intracellular organisms, and we have methods of cultivating viruses which are not commonly used nowadays. But we have different cell lines which you encounter in systemic microbiology, like rabbit cell line, Hep2 cell line, okay, HeLa cell line, K13 cell line. All these are cell lines, right? So either you can have animal inoculation like Coxsackie virus and arboviruses. Okay, Coxsackie virus and arboviruses are inoculated on suckling mice. Okay, Coxsackie and arboviruses on suckling mice. Fertilized or embryonated hen's egg, you can inoculate the virus on fertilized or embryonated hen's egg. Okay, so you can see the embryonated hen's egg where you have four different areas where you can inoculate the virus and grow it, cultivate it, right? You can either grow in chorioallantoic membrane. Which two viruses are grown in chorioallantoic membrane? Pox virus and herpes virus. Both are enveloped viruses, right? Pox virus and herpes virus are grown on chorioallantoic membrane. Amniotic cavity, isolation of influenza virus. Influenza virus is amniotic cavity. Okay. On yolk sac. On yolk sac, we have arbovirus, rickettsia, and chlamydia. Okay, arbovirus, rickettsia, and chlamydia. On allantois, you can do a subculture on the influenza virus. So influenza virus is grown on amniotic cavity, but on allantois, you can do a subculture. Okay. So this is the thing. You can take a screenshot if you want, and let's move ahead now. Okay. Next comes our tissue culture or cell culture, how it is cultured in a cell. So it is the most common method to culture a virus. Animal inoculation raises some ethical issues, right? And on embryonic handshake also not commonly used, but you can do cell culture, primary cell line, diploid cell line, continuous cell lines, right? The primary cell line cells multiply 5 times, okay, like human amnion cell line. And diploid cell line means cell multiplies 50 times, about 50 times, like W138 cell line. Okay, WI38 cell line. WI, it's I. Okay, WI38 cell line. Okay, primary cell line, human amnion cell line, diploid 5 times, then 50 times, WI38 diploid cell line. So these both can be used for viral vaccines. And continuous cell lines when the cells multiply indefinitely. So you can have HeLa, Hep2, McCoy, KB. HeLa, Hep2, McCoy, you just remember. Okay, not used for vaccines. Only viruses that can be seen on light microscopy is pox virus because it is very large. Okay, it is the largest virus. So it is the smallest Papova virus, right? Okay. How to detect the presence of viruses on cell line? How you can know that the virus is present on the cell line, right? So you can have, you can remember this by chimti. Okay, CHIMTI, chimti. Chimti, you know that you use in cooking, right? C is the cytopathic effect, which is the most common one, which is occurring, okay, change occurring in a cell due to presence of virus. How your cells, body cells react to a viral infection. This is a cytopathic effect. Okay, we'll discuss the different type of cytopathic effects like grape cluster formations and all now. Heme adsorption. The influenza virus shows heme adsorption. Viral interference. Okay, viruses tend to interfere with the host machinery, right? So viral interference, metabolic inhibition, transformation, and immunofluorescence. Immunofluorescence is the most common method, right? Most sensitive and specific. All these are there. Okay. So cytopathic effect. Coming to cytopathic effect. Okay. So you can have different types of cytopathic effects which are the virus is not producing the non-cytopathic virus. Okay. Okay. So you can remember this CMT, right? Cytopathic effect. Then H was heme adsorption. Okay. I was the interference. M was the metabolic inhibition, transformation, and your immunofluorescence. Cytopathic effect. You can have inclusion bodies. Okay. So we can have some bodies which are present, like Negri bodies in rabies. You must have heard Negri bodies. So inclusion body either may have intranuclear inclusion bodies or intracytoplasmic inclusion bodies. Okay, intranuclear are formed by DNA viruses and intracytoplasmic are formed by RNA viruses, right? The exception is pox virus. Pox virus forms intracytoplasmic inclusion bodies. Done. And intracytoplasmic inclusion bodies are produced by RNA viruses, or formed by RNA virus. Okay, exception is yellow fever virus, which produces intranuclear inclusion bodies. Okay, so intranuclear inclusion bodies by all DNA viruses, intracytoplasmic by all RNA viruses. What is the exception in here? Pox virus, which forms intracytoplasmic. Okay, pox virus forms intracytoplasmic. And what is the your exception here? Your yellow fever virus. Yellow fever virus produces intranuclear. Okay. So both intranuclear and intracytoplasmic inclusion bodies are produced by cytomegalovirus and measles virus. So measles virus and cytomegalovirus are two viruses which form intranuclear also, intracytoplasmic also. Okay, this you have to remember. So what is the inclusion body of yellow fever virus known as Torres bodies? Okay, just we have a list here of what are the inclusion bodies which are there. Okay, Negri bodies are seen in rabies. Molluscum bodies, molluscum contagiosum virus. We'll look at the molluscum contagiosum virus in time. Guaneri bodies in vaccinia virus, Bollinger bodies in fowlpox virus. Fowlpox virus is not much important. Perinuclear cytoplasmic acidophilic bodies in reovirus not important. Owl-Sai inclusion bodies in CMB. Okay, Owl-Sai appearance. And Cowdrey type A. So we have two type of Cowdrey type A, Cowdrey type B. Okay, Cowdrey type A in herpes simplex and measles virus. Okay, this much is important. So first cytopathic effect was inclusion body formation. Second is syncytium formation. The cells try to cluster in a grape-like cluster. They form the not grape-like. Just I'm telling. It is a multinucleated cell formation. Okay, it is a multinucleated cell formation like paramyxovirus. Paramyxoviruses tend to clump the cells and form a giant cell. Okay, giant cell is being formed. And some herpes viruses like HSV1, 2 varicella zoster, HSV1, 2 varicella zoster form the multinucleated giant cells. Right. Grape-like viruses like adenovirus, shrinking and degeneration of cells, cicorna. Cicorna is something sounding like picorna. Okay, like picorna viruses, the RNA viruses. RNA, you can see here, RNA word is there. Okay, it is RNA virus. So cicorna sounds like picorna. Okay, so picorna viruses, so shrinking and degeneration of cells. So that was our basic discussion on the introduction to virology. So you have to know these all facts. You can remember this. You can just rewind the 16 minutes I took on the introductory part, and then we are gonna shift to the virus proper. Okay. So we'll start one by one and move on. As we move ahead, we'll discuss different viruses. Right. So let's begin on the journey of virology, and let's read about the structures that's the most smallest but cause the dreadliest of infections. Okay. Let's start with the viruses now. So welcome. Here we are starting with our discussion on parvoviruses. Parvovirus B19 is the most significant one. Okay, and talking about so the smallest virus is the parvovirus, and you know this linear ssDNA is present. Okay, ssDNA is present. The smallest virus, non-enveloped, and icosahedral symmetry is there. Icosahedral symmetry, you can just get a pictorial memory of this picture of parvovirus. You can just just see the helical one, icosahedral symmetry. Only one serotype because it's the smallest one, so one serotype. Many viruses have different serotypes, right? Transmission is the respiratory route, transplacentally or blood transfusion, right? So respiratory one is the most accepted route of transmission of this parvovirus. Humans are the only reservoir. Humans are the only reservoir. Pathogenesis. The virus infects erythroblasts and endothelial cells of blood vessels. Okay. Erythroblasts. So it is a parvovirus. P. P is there, right? So it attaches to the P blood group antigen. Okay, you can just remember from the name itself that P means it attached to the P blood group antigen on the erythroblasts or the RBCs. And endothelial cells is partly associated with rashes. Okay, it causes rashes. Immune complexes are formed. Okay, it forms immune complexes and cause rash and arthritis. So one injection gives one infection gives lifelong immunity against the virus. It's like the chickenpox virus also. Okay. Clinical features. Okay, we have to remember the clinical features is the erythema infectiosum. Erythema infectiosum it's called as the fifth disease. Okay, fifth disease. Erythema infectiosum. It is a mild illness. Okay, so don't get confused between different type of erythema. Okay, like many other erythema infectiosum is one of them. Just don't get confused. In further course, we'll read about many erythemas, right? So infectiosum, also known as fifth disease. It is a mild illness. Incubation period is 7 to 10 days. You can remember fifth disease, right? Into 2 is 10. 7 to 10 days, you can remember the range. Mild illness seen in childhood. Bright red maculopapular rash. You can see the picture. Just register this picture also in a pictorial memory in cheeks. So it is known as a slapped cheek appearance. Okay, a slapped cheek appearance accompanied by low grade fever, sore throat, and running nose. So a lacy, less intense erythematous rash occurs in the body. Okay, a type of erythematous rash is basically occurring. It's a lacy in nature. A resolution occurs in 1 week. Okay, most of the parvovirus infections are asymptomatic, right? It is not symptomatic. People with sickle cell anemia, thalassemia, and hereditary spherocytosis develop a transient aplastic crisis. Okay, so this virus is causing a aplastic crisis. Why? Because it is attaching to the erythrocytes, right? So it will cause a aplastic crisis in which people in the sickle cell anemia traits, okay, thalassemia and hereditary spherocytosis patients. Infection in pregnancy is it is a transplacentally transmitted virus. So infection in first trimester the virus infects, then there's a stillbirth, right? Second trimester we can have hydrops fetalis. Hydrops fetalis, there's a massive edema in your brain, right? Massive edema due to congestive cardiac failure precipitated by anemia, right? And infection in third trimester don't have any much significant effects, mild effects, some may be there. Arthritis in adults, it causes arthritis in adults. So majorly we saw in the infectious erythema infectiosum, which is the fifth day disease. Okay, and the incubation period you can remember from that is 7 to 10 days, and it can cause arthritis in adults. Okay, females are more affected than males. Small joints of hands and feet are affected in a bilateral symmetrical manner, resembles rheumatoid arthritis. Some other viral infections which cause immune complexes mediated arthritis. So you can have hepatitis B and rubella also, which can infect the arthritis. Okay, it can cause the arthritis by immune complexes. Okay, and if you have a chronic infection, right, in people with decreased immunity, so most of the people have good immunity, so you can contain the parvovirus infection, small virus, right? Not much infections are there here. So you can have chronic anemia because it is infecting erythrocytes again. Okay, and leukopenia and thrombocytopenia. So basically, you have to remember this fact that it is affecting two things: erythroblasts and the endothelial cells of blood vessels. Okay. Let's come to lab diagnosis. So fifth disease and aplastic anemia are there. Okay, so you can how can detect antibody detection, IgM antibody detection, culture of virus from throat swab. You can take a throat swab. Most common mode is respiratory one. So you can take a throat swab and do the culture, right? And in immunocompromised people, you can do PCR. PCR is basically increasing your viral load, right? PCR basically you can say making more copies of the virus. So you can do PCR in immunocompromised people. And fetal infection is there. You can take a PCR of the amniotic fluid. You can aspirate the amniotic fluid out and do a PCR. What is the treatment? No specific antiviral is there. Pooled immunoglobulins. You have to give immunoglobulins, and no vaccines or prophylactic measures are available here. Okay. So pox virus, just let me quickly go through. It is a linear virus, ssDNA virus, important one. ssDNA, just this is important. Smallest virus, about 20 nanometers, right? Non-enveloped icosahedral. Okay, transmission is by respiratory, transplacental blood route may be there. Pathogenesis infects the endothelial cells and the erythroblasts, right? Tetrasto P blood group antigen, and endothelial cells are responsible for rashes. Immune complexes are formed which causes rash and arthritis. Okay, and the clinical features you can have erythema infectiosum, 50-day disease in the maculopapular rash is the bright red in color, lacy rash. Okay, so we can have a special appearance which is asked, that is slapped cheek appearance, right? And resolution occurs in 1 week. Most of the parvovirus infections are asymptomatic. Okay, they are asymptomatic infections. So in patients with sickle cell anemia, thalassemia, hereditary spherocytosis, you can have a transient aplastic crisis. Transient aplastic crisis, not permanent, right? And if infection in pregnancy, first trimester, just dead. Okay, in second trimester you can have hydrops fetalis, massive congestive edema will be there, and in third trimester you can have no important clinical features. Arthritis occurs in adults, which resembles a type of rheumatoid arthritis, right? So hands and joints are swelled in bilateral symmetrical fashion. Other infections are there with hepatitis B and rubella. Chronic B19 infections, they infect people with decreased immunity, right? And lab diagnosis, you can do fifth disease and aplastic anemia. You can do IgM antibody detection. Okay, you can do a throat swab and cultivate the virus. You can, in immunocompromised people, you can use PCR, and fetal infection, you can do PCR of amniotic fluid. Treatment is no antiviral, pooled immunoglobulins, and no vaccine is available. Okay, so that's what about parvovirus. Let's move on to the next virus. Okay, after the smallest virus is done, we are into the largest virus, right? This is the pox virus. Okay, so pox virus is, just remember, it's the biggest virus, 200 nanometer, about 10 times more bigger than the parvovirus, right? It's a DNA virus which induces intracytoplasmic inclusion body. Okay, intracytoplasmic inclusion body. I told about inclusion bodies. Don't skip the initial part. It's very important, right? Only viruses which can be seen on light microscope due to its large size. Envelope is present. I told you that it is the aristocratic virus. Other aristocratic viruses are the Hep DNA viruses, the hepatitis B virus, and the herpes virus, right? Envelope is present. It is having a icosahedral symmetry. Again, important pox viruses are variola, vaccinia, and molluscum contagiosum. Three important pox viruses you have to remember: variola, vaccinia, and molluscum contagiosum. Right? Variola is the smallpox. Vaccinia virus is basically used as a vaccine of smallpox virus. Right? So if we're talking about the variola virus or the smallpox virus, you can see the structure here. Okay, you can see the structure. There are some ridges which are present. This is the outer membrane, inner membrane, means envelope is present. The core and nucleocapsid, which is 12 nanometer thick. You don't have to remember this, just remember the shape. It's a dumbbell shaped or biconcave shape. Lateral bodies are present in the sides. dsDNA is present. Okay, dsDNA, since it is a DNA virus, all have dsDNA, right? Viral enzymes are present. Symmetry is complicated or complex symmetry as written in many texts rather than icosahedral. We tell it icosahedral for our own simplicity, but the symmetry is a bit complex here, right? Smallpox or variola virus is disease is smallpox, and it is the first infectious disease to have been eradicated from Earth. Okay, it is the only infectious disease which is eradicated from Earth, the first one, since it doesn't have animal reservoirs, so we don't have to fear a zoonotic spread, right? It is not asymptomatic or subclinical cases. Carriers are not, neither there will be no disease or there will be disease. There is no in between state, right? And characteristic clinical features are produced, which you can see here. Okay, a characteristic you can say a leonine type of appearance, right? And it has a very good vaccine also. We have a very good vaccine. So mode of infection is droplet inhalation, just like your parvovirus. Incubation period is 7 to 18 days. 7 to 18 days. Okay. Rashes are deep seated. All rashes are one stage. So slow evolution of rash. The rashes start and slowly evolve. Palms, soles, and extensor surfaces are first affected. So we are involving the palms and soles. Rashes are involving palms and soles. Important statement. Fever subsides with appearance of rash. Right. Then we'll come to the lab diagnosis of this virus. Right. Okay, so in lab diagnosis, we can have detection of intracytoplasmic inclusion bodies, that is called as PASTU bodies. You can remember pox virus and PASTU bodies, right? So intracytoplasmic inclusion bodies are produced by pox virus, right? So you can have PASTU bodies. So you can remember that all DNA viruses produce intracytoplasmic inclusion bodies, right? And we had an expression also, right? Okay. Electron microscopy, you can have brick-shaped morphology of this virus. Okay, the virus is big brick-shaped. You can look at the diagram. It is like a brick. I have drawn it with brown color to make it a evident like it's a brick shape, right? Characteristic pock formation. Okay, just you knew that when we have a viral inoculation on chorioallantic membrane, it produces characteristic pocks. Okay, POCKS on fertilized or embryonated hen's egg. Okay, this type of pock formation is there, right? And treatment, we have vaccinia, immunoglobulins, and antivirals. So vaccinia, immunoglobulins, antivirals like cidofovir and methicillin. Cidofovir and methicillin. Cidofovir is the antiviral. Remember this. Okay. So vaccinia virus has arose from serial subculture of cowpox virus. Okay, cowpox virus have been serially subcultured, and we get the vaccinia virus used in vaccine, and unattenuated live vaccine is made, right? Variolation. What is variolation? People were inoculated with skin scrapings from a smallpox patient to develop immunity. Variolation was a technique of earlier times, in which the healthy people were inoculated with a skin scraping from smallpox patient, right? A skin scraping from smallpox patient is inoculated, which was inducing an immune response, right? Cowpox vaccine was devised by Sir Edward Jenner before the vaccinia virus vaccine. Okay, and now let's move on to the vaccinia virus, very short virus. Earlier, non-pathogenic or mild skin lesions it causes, and the inclusion bodies are called as Guarneri bodies. We learnt it in general features of viruses, right? Guarneri bodies. Okay. On chorioallantic membrane, pox are found. Pox are larger, hemorrhagic, and necrotic. Pox are larger, hemorrhagic, and necrotic than those of variola virus. Sealing temperature is the highest temperature beyond which pox formation is inhibited on CAM. Okay, on chorioallantic membrane, the highest temperature beyond which POC formation is inhibited is called the ceiling temperature. Ceiling temperature of vaccinia virus is more than the variola virus. Okay, ceiling temperature of vaccinia virus is more than the variola virus. How can you remember? Variola has 7 letters, right? 7 letters. Vaccinia has 8 letters, right? Which is more vaccinia. 41 degrees Celsius, and this is 38 degrees Celsius, right? So this is the ceiling temperature at which the POC formation on chorioallantic membrane on embryonated hen's egg is inhibited, right? So we told that here is the POC formation of fertilization embryonated hen's egg, which is inhibited by 41 here and 38 here. Vaccinia virus also induces plaque formation on chick embryo tissue culture. Okay. So how you can differentiate vaccinia versus variola? The first one is the ceiling temperature. Second one is the plague formation. Plague formation. Plague formation occurs on chick embryo tissue cultures. Okay. And now we come to another viruses, a group of virus which is the molluscum contagiosum virus. Okay, molluscum contagiosum virus, which causes the disease molluscum contagiosum. It is a non-cultivable viruses. Humans are the only host here also. What is the characteristics of molluscum bumps? You can remember by P2W. If you don't have a idea of mnemonics, then you can just remember it in a sequence. So you can have pinkish pearly appearing lesions. You can just register this diagram in your memory, and if you're registering this diagram, then you can just visualize how the lesions are gonna occur in molluscum contagiosum. Molluscum means mollusca. Mollusca is a we have read the animal kingdom, and mollusca is there, right? So molluscum contagiosum virus are soft lesions. Okay, they're what like umbilicated lesions. The umbilicated lesions means a projection type of lesion, pinkish and pearly appearing. Lack of inflammation and necrosis, and no inflammation, no necrosis. Inflammation may be associated, but there is no necrosis. Okay, present in singles or clusters, can occur in skin embryo except palm and soles. Remember, except palm and soles. But if we have read the smallpox, what are the description of the smallpox lesions? You can have not lesions or rashes, you can say deep seated. Okay, all rashes of only one stage, no evolution, slow evolution, you can say. Palms and soles are affected, they're first affected in fact, right? And fever subside with appearance of rash. Fever will subside with the appearance of the rash, right? And molluscum contagiosum virus have pinkish pearly appearing wart like umbilicated lesions, lack of inflammation and necrosis, present in single and cluster, can occur in skin anywhere except palm and sole. Genital ulcers may be seen in adults, right? What's the mode of infection? In kids are direct contact and indirect contact, so might bone may be there. Adults have direct indirect and sexual route even. Lesions disappear in 3 to 4 months. They appear for 3 to 4 months only. No systemic complications are associated with it. Sometimes lesions may persist for 5 years. In HIV infected people, we have a decreased immunity. Decreased immunity means there will be more lesions, more severe lesions which last longer. More generalist, more severe, last longer. Okay. What is the lab diagnosis? Again, the cytopathic effect. What's the cytopathic effect? The inclusion bodies. Okay, inclusion bodies. So we have a molluscum bodies or Henderson-Peterson bodies. Okay, molluscum body or Henderson-Peterson body. Again, it is a pox virus. So it is a DNA virus, and DNA virus is intra cytoplasmic inclusion bodies, right? So seen in skin scrapings by doing HP steps. So molluscum bodies are Henderson-Peterson or HP bodies. So HP bodies is a spotter for you. Here you can see the HP bodies here. This type of appearance is the HP bodies. You can do electron microscopy to visualize the virus, and you can do PCR also. Okay. Treatment is surgical removal by cryotherapy or laser therapy. Just a laser ablation or type of ablation is done. Okay, it's the mainstay of treatment. And pseudophobia is also effective. Okay, pseudophobia is effective in the previous virus, also the smallpox virus. So we are done with all the pox viruses. We are done with the parvovirus. We are done with the pox viruses. Let's move on to the next virus. Okay. So welcome, and we are into the another virus, that is the human papilloma virus. Okay, HPV, human papilloma virus. Human papilloma virus is again a non-cultivable virus. Remember the last non-cultivable virus was the molluscum contagiosum virus, right? And it has 100 genotypes. It has 100 genotypes. Non-enveloped and the genome is there. Genome has two type of genes, two group of genes you can say. One is the early region genes, one is the late region genes. Early region genes were E1 to E7, E1 to E7. Late region genes we have L1, L2. Late region genes we have L1 and L2. Early region genes code for non-structural proteins, and late region genes code for structural proteins. Okay. Non-structural proteins means enzymes. E1 to E7 code for enzymes, and L1, L2, that's the late region gene, code for structural proteins. So what is the structure? Capsid. On the minor capsid protein, L1 codes for major one, and L2 codes for the minor capsid protein, right? Papilloma virus, that is HPV, infect the squamous epithelial cells. So it is basically causing a type of squamous cell carcinoma. Okay, so whenever in the pathology, even when you encounter a squamous cell, squamous cell carcinoma, where the what is the pathological spotter for squamous cell carcinoma? Is the keratin pulse, right? Keratin pulse are the spotter for the squamous cell papilloma or carcinoma, I can say, not papilloma. So characteristic cytoplasmic vacuole formation is there, and such vacuolated cells are called the coelocytes. Coelocytes are the hallmark of the inflammation that is occurring due to this papilloma virus. So in papilloma virus, a characteristic cytoplasmic vacuolation is there, and this vacuolation is called the coelocyte. And coelocyte is the hallmark of inflammation, right? So you can see here how it is invading the skin, and the HPV infection is occurring. Okay, whenever there is a wound, it is invading the skin and going deep into the dermis. Okay, and so reaching up to this dermal portion and infecting the cells from the basal layer. Okay, so it's latent this thing is happening, the latent infection, and it is ultimately replicating and going up. Replicating and going up till the keratinocytes. Okay, so in the keratins, the viral assembly and release occurs in the topmost layer. Okay, topmost layer, HPV viruses are produced. Okay, so what are the genotypes of HPV virus? Okay, human papilloma virus. What are the genotypes? So we have the high risk genotypes and the low risk genotypes. So high risk genotypes we have 16, 18. Okay, just remember like this: 16, 18. Okay, these are the even numbers. 31, 33, 35 odd numbers, and 35 means 45, right? 45. No trick is there, just you have to do an intuition, right? 16, 18, 31, 35, 31, 33, 35, 45. No, not difficult to remember, right? So cancer of cervix, anus and penis are caused by the high risk genotypes. Genotype 16 is responsible 50% of all cases of carcinoma cervix. Okay, so carcinoma cervix, the most common genotype infection is genotype 16, and it's also associated with oral cancers even. Genotype 18 is responsible for about 20 to 25% of carcinoma cervix, right? So basically 80 to 85% of carcinoma cervix is caused by this virus, HPV 16 and 18 genotypes, right? HPV, human papilloma virus, is transmitted by either direct contact or by sexual route, right? So it is a sexually transmitted disease. So E6 protein, what it does, it breaks down the P53 protein, and E7 breaks down the RB. Okay, these are the tumor suppressor genes. Okay, so whenever the E6 and E7 are binding and breaking down the proteins encoded by the tumor suppressor genes, so there is, you can say, there is a loss, inducing a loss of function in this. Okay, so whenever there is a loss of function mutation, okay, or is a loss of function of protein in the tumor suppressor gene, the tumor will develop. Okay, the tumor will develop. So there is an induction of cancer. Okay, easy concept. Warts by this HPV are mostly benign and don't progress to cancer. Both CMI and antibody induced by are induced by HPV infection, helping the spontaneous resolution of warts. In immunocompromised people, more extensive warts are present. So basically, it is causing a wart-like disorder. Okay. Females with HIV infection, very high rate of carcinoma cervix. Okay, the females in HIV infection, very rate of carcinoma cervix. The skin and plantar warts are caused by HPV 1, 2, 3, 4. 1, 2, 3, 4 genotypes are causing skin and plantar warts. Anogenital warts are called as the condyloma acuminatum. Okay, it is if I have read pathology, I must have read about this condition. It's a precancerous condition. Okay, and it's the anogenital warts called as the condyloma acuminatum. Okay, it develops on the skin, mucous membrane of external genitalia, and perianal region. On the external genitalia, perianal region, transmitted sexually, affect males and females. Most common genotypes are 6 and 8, 11. Okay, 6 and 11. And the pregnant lady on vaginal delivery may pass on to the virus to the baby, and here in the baby it causes recurrent laryngeal papillomatosis. Okay, it causes in the baby it causes recurrent laryngeal papillomatosis by genotype 6 and 11. Okay, so flat warts or verruca plana are seen in kids. Okay, what type of warts are seen? Verruca plana. Okay, or flat warts. Common warts are known as verruca vulgaris. Verruca vulgaris is seen in older kids. In kids, smaller kids, your verruca plana is there, and a verruca vulgaris is seen in older kids and occurs on basically the bony prominence, over the bony prominences most commonly, and this is seen on cheek. Okay, this is seen on cheek. Okay. Carcinoma cervix. So, what are the pre-malignant conditions of carcinoma cervix? So, basically, this is a normal cervical epithelium. Okay, if there is a mild dysplasia which is involving one third of the thickness of the epithelium of the cervix, then this is CIN1. Okay, only mild one. Then CIN2, which involves about two third. Then CIN3, which is involving almost all. Okay, it's not involving a few ones, you can see it almost involves almost all of the epithelium. And carcinoma in situ, there is a CIS, involves the full thickness of the carcinoma cell. And this is the invasive carcinoma, means still this the basement membrane hasn't been invaded, but here the basement membrane is invaded and it is causing the cervical cancer. This condition is the cancer. Okay, these all are precancerous condition up to CIS. Okay. Okay, so we told you that the first complication is your anogenital warts, that's the condyloma acuminatum. Look at this, condyloma acuminatum. Condyloma acuminatum, it passes to the baby, then causes verrucous warts. Verrucous warts are the, your plain warts or flat warts. Okay, verruca plana. Verruca plana. In older kids, it causes verruca vulgaris. Okay, verruca vulgaris. Okay, and then we have something called as the epidermodysplasia verruciformis. So there are many verruca terms associated with this infection, the HPV. HPV is an important virus. Please do pay attention to my words. So epidermodysplasia verruciformis is a rare autosomally recessive condition. It is a rare autosomally recessive condition. Increased risk of squamous cell carcinoma. Okay, this seen with genotypes 5 and 18. Your verruca plana and recurrent laryngitis, that is the recurrent laryngeal papillomatosis. Okay, this is seen with genotypes 6 and 18. The anogenital warts, the condyloma acuminatum is associated with 6 and 11, right? And this recurrent laryngeal papillomatosis is also associated with 6 and 11. Okay, and this epidermodysplasia verruciformis is associated with 5 and 8. 5 and 8. Okay, so HPV can also cause pityriasis or versicolor like lesion. Pityriasis is a versicolor like lesion. Immunocompromised people, like organ transplant recipients, it can cause pityriasis. Okay, what is the lab diagnosis of HPV? So molecular diagnosis to detect the DNA of HPV. So HPV, human papillomavirus, is a DNA virus, right? So we can have PCR or hybrid capture assay. Important. PCR or hybrid capture assay. What are the targets? E6 and E7 genes are the targets. Using molecular method, you can predate the cancer. Screening is done by Pap smear. Pap smear was is called the Papil-Nicolau test. The device was George Papil-Nicolau. Right. We can have a conventional technique. We can have a liquid type of Pap smear where you use in the conventional you use air spatula. Okay, spatula is there, air spatula, which is used to take a and put it in a solution, right? And Pap smear can have different type of cells. They are like basal cells, intermediate cells, superficial cells. Okay, you can make a diagnosis. This is the pathological aspect. I will cover in some other video if time permits, right? Confirmation is HPV, HPE or biopsy specimens. Okay, you can do a histopathological examination. So this is more kind of pathology thing. Okay, treatment. Genital warts are treated by podophyllin. Okay, podophyllin. Alpha interferons are effective for preventing recurrences. For skin wart, you can do liquid nitrogen therapy. Okay, liquid nitrogen is used. For plantar warts, the painful warts on the foot. Okay, you can do surgical excision. Basically, surgical excision is done. And salicylic acid tropically. You can apply salicylic acid tropically. Cidofovir is here useful here. So we have some HPV vaccine, which is important because HPV vaccine has gained a good importance over here. Okay, so you can have a subunit vaccine, a bivalent, a quadrivalent, and a nonavalent vaccine, a nine-valent vaccine. Okay, to remember all this. Okay, and the serotypes which they are targeting also. So subunit vaccine is made up of okay. Okay, I was talking about subunit vaccine. Subunit vaccine is containing a virus-like particles, okay, made up of L1 protein. L1 protein. Okay, position east by recombinant DNA technique. L1 protein was a structural protein or non-structural protein? A structural protein. Okay, it is an early protein or late protein. L means late protein. What it is coding for? For the major capsid protein, right? L1 is the major capsid protein. It's targeting the major capsid protein. Bivalent vaccine, which is called Cervarix. Quadrivalent is called Gardasil, and nonavalent vaccine, 9-valent vaccine is called Gardasil 9. Okay, Cervarix, Gardasil, and Gardasil 9. Okay, three vaccines. So Cervarix is targeting 1618 only, 1618. Gardasil is targeting 1618 611, 1618 611. So it can be used in this Cervarix. You can give in your condyloma acuminatum. Condyloma acuminatum was caused by 6 and 11, right? So you can give here Gardasil. Okay, Gardasil. Gardasil can be used in 618 611. Okay, 1618 611. And Gardasil 9. The Gardasil 9 is a recent one. Okay, which is given IM. Okay, 2 doses in 6-month gap between 9 to 14 years. Then 3 doses 0 to 6 months between 14 to 26 years. Okay, so you can have 16 18 6 11, 31 33 35, 31 33 45, and 50 to 58. Just remember in this sequence: 16 18 6 11, 31 33 45. 35 you excluded. 50 to 58. 50 to 58. The Serum Institute of India have introduced Servavac. Servavac is most commonly given in educational institutions and all. 16, 18, 6, 8, 6, 11. Okay. Only it is like the quadrivalent vaccine or the Gardasil only. Okay. Servavac 6, 11, 16, 18. It is again a subunit vaccine. Okay. It contains L1 protein of all above genotypes. It contains the L1 proteins of 6, 18, 16, 18 of all the genotypes. Okay. So that was about our human papilloma virus. We done about your what virus you covered? Parvovirus. We did about pox viruses. We discussed about papilloma virus. Okay. Now we'll move to the next virus. So just remember, another important point is that some viruses causes UTI, urinary tract infections. Okay, what are the viruses causing UTI? We have the BK virus, okay, BK virus and adenovirus. Adenovirus, you have to remember UTI, right? UTI. I looks like 1, right? So 11, 21. 11 and 21 serotypes, they are causing you urinary tract infections. So just I had a point to tell from genetic urinary. You have to just remember this. Okay, let's move ahead. Okay, so we are starting with a big virus, a group of viruses specifically. We have herpes virus. So 8 viruses are there. Okay, so we have to discuss all the 8 viruses one by one, and you have to retain it also. Okay, so I know it's a difficult task, but we'll do it. Herpes virus. So herpes virus. You just take a sip of your favorite coffee and drink a glass of water, and we are gonna start with this part. Okay. So herpes virus, we are discussing here herpes virus 1 and 2. Then we'll move to herpes virus 3, 4, molluscum contagion and done, right? Cytomegalovirus and all other Epstein-Barr, all other. Okay. Herpes viruses, first one is the DNA virus, right? This is second largest overall after pox virus. Pox virus size 200 nanometers. Is it visible by light microscope? Yes. Which is the smallest one? Parvovirus. Parvovirus, which is more medically significant one, B19. What is the size? 20 nanometers. Okay, are done with all? You can answer all the questions? Yes. Okay. Dictum is once infected, always infected. If you are infected with herpes virus, if you are once infected, then you are always infected with the herpes virus. Okay. They set up latent infections. So we have icosahedral symmetry again. Envelope is present, which has peplomers. Okay, as you can see, your peplomers are present. Envelope. It is a dsDNA virus. Again, dsDNA virus. Okay, come under happy viruses, right? Yes. Size is 120 to 200 nanometers. Okay, so 200 nanometer was the size of your pox virus. Okay, so 120 to 200 nanometers is the size of the herpes virus. This is the electron microscopy picture. So you can have 8 viruses in herpes virus. Herpes virus 1 is the herpes simplex virus, HSV1. Second is the herpes virus 2, no specific name. Third is the herpes virus 3, varicella zoster virus. BZV is the herpes virus 3. Herpes virus 4 is the Epstein-Barr virus. Epstein-Barr virus. You can remember bar, BARR, 4 letters, right? 4. So Epstein-Barr virus is the herpes virus 4. 5 is the cytomegalovirus. Cytomegalovirus, CMV. Okay, it's called herpes virus 5. 6, 7 have no common name. 8 is called as the Kaposi sarcoma virus, KSHB. Kaposi sarcoma associated herpes virus. Kaposi sarcoma, you must have heard disease in pathology. Okay. Let's review the names. Herpes virus 1, herpes simplex. Herpes virus 2, no specific name. Herpes virus 3, which virus is varicella zoster virus. Herpes virus 4, your VZV. Varicella zoster is herpes virus 3. Herpes virus 4 is the Epstein-Barr virus, right? Herpes virus 5 is the cytomegalo. 6, 7, no specific name. 8 is Kaposi sarcoma. Done? Yes. They replicate in nucleus and form intranuclear inclusion bodies. They form intranuclear inclusion bodies. Only viruses develop their envelope by budding from nuclear membrane. I told you that enveloped viruses they tend to bud from the host cell membrane. Tegument is present. Okay, so this is the structure. You can see there's a tegument, there's an envelope. So tegument is present between the nucleocapsid and the envelope. Okay, and tegument has certain regulatory proteins which play an important role in viral replication. I told you in the introductory part also. So starting with HSV1, herpes simplex virus 1. The most common clinical feature of primary infection is called gingivostomatitis. Gingivostomatitis. You can see the diagram. It may occur in the gums also, and it may occur in the tongue also. Okay, this type of swollen, painful gums or where some vesicular lesions occur in the mouth. In the oral cavity, they cause some vesicular lesions. Recovery is followed by latent infection in trigeminal ganglion. Okay, so once infected, always infected. Basically, the viruses will resolve. Okay, will recover. Your mouth will be like swollen, and your gums will be swollen, and vesicle lesions will be there. Okay, but you will recover. You don't know that you are infected with herpes simplex, but there is latent infection in the trigeminal ganglion. Okay, you have anatomical description about trigeminal ganglion in your head, right? Okay, so there is a latent infection occurring in the trigeminal ganglion. Okay, and reactivated by the trigger of fever, sunlight, stress, menstruation, immunocompromised state. Remember, whenever there is an infection, okay, and immunocompromised state always precipitates the infection. Just remember this. Okay, fever, sunlight, stress. Okay, this type of any type of stressful situation basically is called reactivation, and when it is reactivated, it causes herpes labialis. Okay, herpes labialis, labia means lip. So herpes labialis, the reactivation infection, right? Here this type of infection is occurring. Other, it can cause herpetic whitlow, herpetic keratitis, herpetic encephalitis. Okay, whitlow, keratitis, encephalitis, herpetic ones. Okay, this type of infections. Whitlow is the occupational diseases of nurses and dentists. Okay, pustular regions of skin of fingers and hands. Okay, basically, Whitlow is basically a nail infection. Okay, fingers, hands. So this is a nail infection, not much severe. Herpetic keratitis. Okay, eye infection. This type of infection, herpetic keratitis, and this is the herpetic Whitlow I was talking about. Herpetic encephalitis, in which the temporal lobes are infected and it is treatable. Okay. So HSV1 most common cause of sporadic encephalitis worldwide. Okay, most common cause of sporadic encephalitis worldwide. HSV1 is transmitted by oropharyngeal contact with infected saliva, salivary contact, and direct skin contact. Basically, it's causing infection of mouth legs, right? So you can just remember this. Salivary contact is important. HSV replicates fast, spreads fast, cytolytic, slow latency nerve cell, belonging to alpha family of herpes viridae. HSV1 cause infection above the waist, HSV above the waist, and HSV2 causes infection below the waist, basically in the genital infection. So HSV1, remember, herpes simplex means simple infections like gingivostomatitis. It is not simple infection. So basically, latent infections are there. Okay, many complications are there, but the initial infection is single one, right? Gingivostomatitis. Okay, and then it enters a latent phase. Latent phase is occurring in the trigeminal neurons, right? And reactivation. Reactivation may be occurring due to sunlight, due to stress, due to menstruation, due to immunocompromised state, okay, due to fever. So basically, in this type of conditions, the infection is progressing to a herpes labialis. Labialis means ulcers are basically occurring beneath the mouth, right? Herpes labialis, you saw the diagram, right? Yes. And we can have three other complications like herpetic whitlow, herpetic encephalitis, herpetic keratitis. So whitlow is infection of hands and this type of fingers. Okay, keratitis infection of eye and the cornea, and encephalitis may cause temporal lobe involvement, and it is treatable. Okay, done. Okay. So now we are into the HSB2. Okay, it is causing infection below the waist, known as the genital herpes. Genital herpes. Most common is the sexual mode of transmission. Vertical transmission is always possible, right? Clinical features is causing multiple genital vesicular lesions. So you can see here this type of vesicle is small vesicles are formed in the, we retract the foreskin, then in the penis itself small vesicular lesions are formed which can break up and cause multiple genital ulcers, and these are painful. It is not that it's painless ulcers, the painful inguinal lymphadenopathy is also observed, and the patient will present with fever and malaise. Okay, then we'll talk about the site of latency. Okay, so if we're talking about the site of latency, the site of latency in HSV1 was the trigeminal ganglion, right? So here the site of latency is lumbar and sacral root nerve ganglion. Trigeminal ganglion is present in the upper above the chest, right? So above the chest infection. The lumbar and sacral nerve ganglion are present below the chest. So there will be site of latency in the lumbar and sacral nerve root ganglion. Same triggers again: fever, sunlight, stress, menstruation, and immunocompromised. 5 triggers. Reactivation infection we call recurrent genital herpes. So the patient first develops genital herpes. Genital herpes is multiple genital vesicular ulcers which are vesicular, filled with clear fluid. It break up and can cause multiple painful ulcers also, and patient present fever and malaise. Okay. Most common cause of genital ulcer disease in the West is HSV2 only. Okay, and site of latency is the lumbar and sacral nerve root ganglion, and triggers are there. Then recurrent genital herpes may set in. Reactivation infections are generally less severe and associated with decreased viral shedding. Genital herpes may be caused by HSV1 also, okay, due to orogenital intercourse. Okay, we can have something called the neonatal herpes. Okay, we can have something called the neonatal herpes. So, what is neonatal herpes? Because the passes the baby through infected birth canal, the herpes virus may be transmitted to the newborn. Okay, you can have neonatal herpes, not transplacentally. So, clinical features may range from severe disease, that is disseminated lesions, to mild local infections. It can have a variety, like it may be asymptomatic even, right? Most common cause of neonatal herpes is HSV2. Important. So you can see what are the diseases by HSV1. It is causing basically above the chest infections. Encephalitis, conjunctivitis. So first we have to remember gingivostomatitis. Then it progresses to herpes labialis. Okay, labialis is there. Then you can have whitlow. Okay, it can have pharyngitis, esophagitis. Okay, then tracheobronchitis, pharyngitis, esophagitis, trachobronchitis. Okay, this type of encephalitis, keratitis, conjunctivitis is there. And in HSB2 may cause meningitis. Just this is one you can remember exception, you can say. Okay, but majorly it causes perianal herpes, genital herpes, and herpetic whitlow also. Okay, herpetic whitlow also. It's a controversial one. Okay, so this is an important one that is called the molarets meningitis. Okay, just remember the molarets. Okay, molarets. Molarets meningitis. What is this? This is a type of aseptic meningitis. I am sure you have read about all the meningitis. Okay. Due to retrograde movement of virus to spinal cord, okay, if the virus retrogradely moves to the spinal cord, then can have this lymphocytic meningitis, chronic headache. The patient will present with chronic headache, right? Diagnosis will be a sample from the lesion. You take a sample from the lesion and you make a smear called as the Zank smear. Okay, you make a smear known as the Zank smear. Okay, and this Zank smear is basically it will be positive. It will produce the vesicular lesions in HSV1, 2, and BZB also. That is the herpes virus type 3. So in Zank smear, you will see multinucleated giant cell formation with intranuclear inclusion bodies. Okay, you take a sample from the lesion. It is called the Zank smear. Prepare a Zank smear. Okay, and the Zank smear you will see multinucleated giant cells with intranuclear inclusion bodies. Definite diagnosis is done by culture, but culture is time consuming and requires more manpower. For herpetic encephalitis, you can do DNA PCR of the CSF sample, right? For your molarets meningitis also, molarets meningitis, you can do a CSF examination, right? What is the treatment? You can use acyclovir and trifluridine. Acyclovir and trifluridine. Trifluridine is basically used in cases of this eye infections due to HSV1. But acyclovir is a good one. Acyclovir is used in HSV1, that herpetic encephalitis. It is also used in systemic diseases also by HSV1. Primary and recurrent genital herpes and neonatal herpes also you can use acyclovir. Okay. So here we are done with the herpes virus 1 and 2. Now we'll move to the third herpes virus, that is the VZV, that is the varicella zoster virus, okay, or the chickenpox virus, right? So varicella zoster virus is known as the human herpes virus type 3. Okay, only one serotype is there. Only one serotype is there, right? And two diseases are caused. One serotype, two diseases: varicella or chickenpox, which is the primary manifestation. Zoster singles is a reactivation manifestation, right? So herpes virus tend to reactivate, tend to cause latent infections, right? Reactivation. So varicella or chickenpox is the primary manifestation, and zoster singles and the reactivation manifestations occur in the sixth decade of life. Sixth decade of life, late presentation is there. Humans are the only host. Virus is transmitted by respiratory droplets and direct contact. Virus infects the mucosa of upper respiratory tract via blood. It moves to the skin and causes typical vesicular rash. Okay, so basically traveling from the respiratory epithelium, upper respiratory tract, and via the blood, it is causing the typical vesicular rash. Okay, so clinical features of varicella, you can look at the chickenpox, have seen. So 14 to 21 days is the incubation period, 2 to 3 weeks. Okay, followed by prodrome of fever and malaise. After 2 to 3 weeks, there's a prodrome of fever and malaise. A papulovascular vesicular rash on trunk and then spread to head and extremities. Okay, a papulovascular rash is there is produced on the trunk and then spreads to head and extremities. Okay, so rash basically evolves from papules to vesicles to pustules to crusts. This evolution is important. Okay, from papule to vesicle, means the vesicle. Okay, papule is just a little projection, vesicle is a fluid projection, pustules are pus filled projections, and after pustules there is a crusting. Okay, so we have a crusts. So papules to vesicles to pustules to crust. Okay, so here there is not a macular papillary rash, there is a papulovascular rash. Okay, first appears in the trunk lower region, then it spreads up to the head and extremities, right? In adults, chickenpox is not self-limiting. 5-day acyclovir is needed. Increased chances of complications are there in adults, but in children it is self-limiting, right? What are the complications? So most common complications is secondary bacterial infection, right? Cutaneous complications. Basically, the skin is very dirty looking in chickenpox because many secondary infections tend to set in. Okay. Most common extracutaneous complication is encephalitis. It may travel to the brain also. Okay, it's a very rare condition. Most severe complication is pneumonia. Okay, Reyes syndrome may occur. Reyes syndrome is seen with aspirin, the type of aspirin-induced one. But here it can show Reyes syndrome. Recovery from chickenpox is followed by antibodies against BZB and lifelong immunity against chickenpox. So once infected with chickenpox, we tell that we don't usually get affected with chickenpox again. But zoster can occur in sixth decade due to decreased immunity. Due to decreased immunity or immunocompromised state, zoster can still occur in the late one, in the late presentation, in the sixth decade of life, right? So what is zoster? Zoster is also called singles. So painful vesicles are formed along the course of a sensory nerve. So basically the a neural spread is basically occurring here of the virus. The reactivation infection, zoster is a kind of reactivation infection. This painful vesicles are produced along the course of a sensory nerve, along the course of a sensory nerve on head or trunk. Site of latency of virus after recovery from chickenpox is the spinal nerve root ganglia. Okay, the site of latency of virus after recovery from chickenpox. After a person is recovered from chickenpox, the virus is basically latent in the spinal nerve root ganglia. Okay, T3 to L3. So this may lead to your singles or zoster. Okay, T3 to L3, anyone. Zoster rash doesn't cross midline. Zoster rash will never cross midline. Pain can last for weeks even. Okay, pain can last for weeks. So it is called the post-zoster neuralgia or post-herpetic neuralgia. Post-zoster neuralgia or post-herpetic neuralgia. In immunocompromised people, varicella pneumonia can occur as a complication, right? Okay. So what is the lab diagnosis? Again, Jank smear. Jank smear we talked about, remember, in HSV 1 and 2, right? So smear made from vesicular fluid, which is stained with GM site, produces intranuclear inclusion bodies with multinucleated giant cell. Multinucleated giant cell with intranuclear inclusion bodies, which is diagnostic of herpes infection. You can do cell culture. Jank smear is positive in which conditions? HSV 1, 2, HSV 2, and HSV 3. These three can show your Jank smear. Jank smear means multinucleated giant cell form with intranuclear inclusion bodies. Treatment is no antiviral to people with good immunity. You don't need to give antivirals. Adult with chickenpox, if you're an adult with chickenpox, 5-day acyclovir regimen is given. If it is resistant, you can use foscarnet. Okay, foscarnet may be used in resistant cases of acyclovir, right? So that was about varicella zoster virus. Let's move on to the next herpes virus, that is the Epstein-Barr virus. That is the herpes virus type 4, right? So we are now moving to the Epstein-Barr virus. Okay, so after the three herpes viruses, we are into a Epstein-Barr virus. That's the herpes virus type 4. Okay, herpes virus, human herpes virus 4, HCP4. So Epstein-Barr B is, I have written bigger because it is a B cell virus. It's in fact the B cell. By attaching to, you have read CD21 or CR2 receptor. CD21 receptor we told in the introductory part is also called the CR2 receptor. Okay, so it is attaching the CD21 or CR2 receptor, leads to malignant transformation of B cells. B cells are malignantly transformed, right? Those are genome of EBV. Okay, the EBV is basically infecting. It means that it's injecting its genome, right? So it is causing a malignant transformation of this. Humans are the only host. It is a ubiquitous virus. More than 90% adults have antibody against the virus. Okay, so basically we must have suffered from any about 90%, 9 out of 10 people have been exposed to this virus earlier. Okay, so transmission is by infected saliva, sexual contact, blood transfusion, and bone marrow transplant. So sexual route may be there, blood route may be there, or infected saliva may be there. So initial application of virus occurs in the epithelial cells of oropharynx and parotid. Okay. So basically, the replication of virus is occurring. It's a respiratory kind of virus. Okay. So the initial replication is occurring in the orthopharynx and the parotid gland. Okay. Then B cells are infected where EBV exists in latent state. Okay. So EBV exists in a latent state on the B cells. So let's see the peripheral blood picture of the EBV infection. So you will see atypical lymphocytes here. Atypical lymphocytes. There are TC cells only. The TC cells only, which are reacting against the virus-infected B cells. So whenever there is an infection in our body, the body tend to react widely. The body tends to react weirdly. So basically, the TC cells are reacting in a different kind of manner, which is giving a appearance of atypical lymphocytes. Okay, so most infections are asymptomatic because we have been exposed and 90% of people have antibodies against this virus. Okay, so these are the images of the atypical lymphocytes. I will tell about it in bit later. The first disease caused by this virus is infectious mononucleosis, also known as IMN, also known as glandular fever. Okay, glandular fever. Why glandular fever? Because the lymph nodes are enlarged, right? So it is called infectious mononucleosis, IMN, or glandular fever. Incubation period is 4 to 8 weeks. Acute self-limiting illness lasts for 2 to 4 weeks. Okay, so incubation period 4 to 8, but it lasts for 2 to 4. Okay, just half. Okay, characterized by fever, sore throat, lymphadenopathy, splenomegaly, anorexia, lethargy, hepatitis, encephalitis, rash. Okay, Parsal, Parsal, you can Parsal, you can remember. It's just anything, do anything, but remember this one. Okay, you can know this grossly also, not in this sequence you require. Okay, so fever, sore throat, lymphadenopathy will occur. Then normal symptoms: fever, sore throat, lymphadenopathy. Splenomegaly will occur. Splenomegaly rupture of spleen is a rare but feared complication of EBV infection also. Anorexia and lethargy, the patient will lethargic, the patient will feverish and all. Hepatitis may lead to subclinical hepatitis, encephalitis, and rashes. Peripheral smell will show atypical lymphocytes. Okay, just remember that this infectious mononucleosis is also called as the kissing disease. Kissing disease. Why kissing disease? Because it is seen in young adults and adolescents who tend to involve in kissing activities. Or this is a saliva transmitted infection. I told you that the virus is basically being transmitted using the saliva. You can see here where I had written. Okay, infected saliva, sexual contact, blood transfusion, and all. Okay, so due to this infected saliva transmission, it is called the kissing disease, right? Okay. Patient serum is positive for heterophile antibody. Okay, so this person will give you heterophile antibody positive test. Okay, heterophile antibody positivity test. Okay, so IMN will show presented with exudative pharyngitis, big lymph nodes, a big spleen, and very atypical lymphocytes. So you can remember by EBV also. If I'm not using the yellow color because this is not much prominent. You can remember by EBV. Okay. EBV, E stands for the exudative pharyngitis. Exudative pharyngitis. Okay. B stands for the big lymph nodes. Big lymph nodes. The lymph nodes are very big. Okay. The big lymph nodes and big spleen. Big lymph nodes and big spleen. B is a very atypical lymphocyte. Very atypical lymphocytes. Okay. These things you have to remember. Okay. Other infectious causes of IMN. What other infections can cause IMN? Just remember CMV, the cytomegalovirus can also cause your IMN, that is infection mononucleosis. Adenovirus can also cause CMV is the herpes virus type 4. Human herpes virus, CMV is the type 5, right? Adenovirus may cause Toxoplasma gondii may cause IMN like symptom. HIV, HSV, and Ehrlichia also. HIV, HB6, and Ehrlichia also, right? All these are heteroviral antibody negative. So other non-infectious causes, any malignancy may cause infectious mononucleosis or autoimmune disorders may cause infectious mononucleosis. So which diseases cause infectious mononucleosis? What viruses also? HSV6, HSV5, that is cytomegalovirus, adenovirus is causing. Okay, and Toxoplasma gondii is causing, it's an opportunistic infection in HIV patient. So HIV is causing. HIV means your Ehrlichia. Okay, Ehrlichia and HIV are related. So Ehrlichia is causing. So this completes our first disease, that is the HIV, which is called the kissing disease. And these are the symptoms: fever, sore throat, lymphadenopathy, and splenomegaly, anorexia, lethargy, and hepatitis encephalitis rash. Right? Okay. Now comes another disease, which is a Duncan disease, which is the X-linked lymphoproliferative syndrome. X-linked lymphoproliferative syndrome is a Duncan's disease. Okay, Duncan's disease. So Duncan's disease is a fatal form of IMN. Okay, it is a fatal form of IMN, infectious mononucleosis. It is a Duncan disease. Okay. It's a very severe in kids with inherited immunodeficiency disease. So it is X-linked lymphoproliferative syndrome. It's also called in kids. It's X-linked. Okay, it is X-linked lymphoproliferative. So you can know that the T cell and NK cells are basically proliferating, and the mutation is occurring in a gene encoding for signal transduction protein. It is for optimum functioning of NK and T cells. So basically, our defense mechanism is basically injured by T cells and NK cells. Okay, are basically not acting much. Okay, due to the gene mutation in gene encoding for the signal transduction protein. There is a mutation in gene encoding for signal transduction protein, due to which there is no optimum functioning of NK cells and T cells. Right. So this is a Duncan disease. Case fatality is 75%. Treatment is bone marrow transplant, cord blood transplant. This is like a good transplants. The severe form, right? So Duncan is a fatal form of IMN. Then you can have third disease. The first one, which disease? Infectious mononucleosis or glandular fever. The second one is the Duncan disease. It's a severe form of infectious mononucleosis. The third one is called the oral hairy leukoplakia. Oral hairy leukoplakia. It's a whitish non-malignant lesion. You can see the lateral surface of the tongue, basically the lateral side of the tongue. This is a whitish non-malignant disorder with a irregular hairy surface, right? Leukoplakia. Oral hairy leukoplakia. OHL. Okay, you can see a white hyperkeratotic plaque usually distributed along the lateral border of the tongue. So this is the lateral border of the tongue, right? It may be associated with HIV also, plus in immunocompromised people usually, and caused by Epstein-Barr virus. What are the cancers associated with EBV? So most commonly, cancers are associated with EBV, like Burkitt's lymphoma, Hodgkin's lymphoma, nasopharyngeal carcinoma. Hodgkin's, Burkitt's, and nasopharyngeal carcinoma, right? So, nasopharyngeal carcinoma has a HLA predisposition, HLA-A2-A8, occurring nasopharyngeal carcinoma, HLA-A2-A8. Okay. The fifth disease, the post-transplant lymphoproliferative disease, PTLD. Okay, not much to learn, but PTLD you can learn. PTLD is the most common presentation of this post-transplant lymphoproliferative disease is the B-cell lymphoma. Okay, so basically Epstein-Barr virus is causing diseases of the B cells, right? So it is basically the PTLD is basically a B-cell lymphoma which occurs after bone marrow or solid organ transplant, right? The main predisposing factor is immunosuppression. What are the diseases? You can remember born to do PhD, Burkitt's, oral leukoplakia, nasopharyngeal cancer. P stands for PTLD, H stands for Hodgkin's, and D stands for Dunker's disease. So I'm repeating once again. The first one was the infectious mononucleosis, most common. Okay. The second one was the Duncan syndrome, it's a X-linked lymphoproliferative disorder, right? And the third was the oral hairy leukoplegia. Okay, it may cause some cancers. Cancers like which type of cancers? You can have the nasopharyngeal carcinoma. You can have the Burkitt's lymphoma, right? Burkitt's lymphoma, Hodgkin's lymphoma. So not mixed cellularity type. Here I had written. It can cause a mixed cellularity type of Hodgkin's lymphoma. I will read in pathology. And the fifth disease is the post-transplant lymphoproliferative disease, that is the PTLD. Okay. Next we move to the lab diagnosis, right? So you can take specimens. What are the specimens you can take? It is causing an infection of your saliva, right? Saliva. So saliva you can take, right? You can take peripheral blood also. You can take lymphoid tissue also. Lymphoid tissue, why? Because the B cells are being infected, right? And then isolation culture of virus. Isolation is a tough and time consuming process, 6 to 8 weeks. Special facilities are needed, not done for routine diagnosis, right? Detection of EBV specific antibodies by ELISA, IIF, CFT, and gel diffusion. ELISA is enzyme linked immunosorbent assay. IIF is indirect immunofluorescence. CFT is complement fixation test, and gel diffusion. Okay. Current infection, increase in viral titer to one of the EBV specific antigen or IgM antibody to viral capsid antigen is occurring. Okay. Early antigens, if the early antigens are there, then it can indicate current infection, Burkitt's lymphoma, or nasopharyngeal carcinoma. Antibodies to, okay, this is important. Antibodies to Epstein-Barr virus nuclear antigen EBNA. Okay, EBNA is commonly asked in your exams. So it indicates past infection. Remember, it indicates past infection. And if there is a very significant increase in titer, it can indicate a current infection, even, right? Or you can have heterophile agglutination, okay, which agglutinate the CYP-RBCs, okay, EBB, and what you are doing is basically you are the EBB contains its antigen, right? EBB antigen is present on the CYP-RBC also. You are putting the same antigen, you are placing the same antigen, okay, and you are mixing the serum and the CYP-RBC and detecting it, and it's detected by Paul-Brunel test, okay. Paul-Brunel test is used for detection. So inactivated serum in doubling dilutions is mixed with equal volume of 1% CYP-RBC. This 1% CYP-RBC you will get from your commercial. You can get commercially, right? So inactivated serum in doubling dilutions is mixed with equal volume of 1% CYP-RBC, incubated at 37 degrees Celsius for 4 hours. A titer of more than 1 is to 256 is suggested of IMN. Okay, a titer of more than 1 is to 256. This is suggestive of IMN. Okay, so Paul-Brunel test, you are basically what you are doing, you are basically taking inactivating serum. The inactivated serum is there in doubling dilutions, diluting double in doubling dilutions. It is mixed with 1% CYP-RBC, 1% CYP-RBC, right? And you are putting in the 37 degrees Celsius for 4 hours. For 37 degrees Celsius for 4 hours, you are incubating it. Then you are taking the titer, seeing the titer, and if it is more than 1 is to 256, suggestive of IMN. But if you have to confirm it, this is just a detection one. This is a screening test. This heterophile antibody screening test. But you have to confirm it, right? So confirmation is done by agglutinin adsorption test, AAT, agglutinin adsorption test. This is important. You can do this agglutinin adsorption test on either guinea pig kidney, guinea pig kidney, or ox RBC. Just pronunciation is incorrect. It's called guinea pig, right? So guinea pig kidney or ox RBC. These are two places you do agglutinin adsorption test, and it is then repeated on Paul Brunel to confirm the result. So what is happening is that basically first you are doing Paul Brunel. Then for confirmation you are doing and your agglutinin adsorption test, AAT. AAT can be done on two cells, or guinea pig or ox, guinea pig. Genepic one, or ox RBCs? Ox. Okay. For confirmation, again you are doing Paul Brunel. This is the sequence, right? Normal serum. What is occurring in the normal serum? So normal serum, the genetic kidney is adsorbed. Okay. On ox services, it is not absorbed. Okay. And on repetition, there is the serum is prior treated. It is adsorbed on genetic. Okay. So on Paul Brunel test, it is so negative result as it is adsorbed. Okay. How will appear in serum? Or in ox RBC, as it is not adsorbed. Just a minute. So it's a busy day. Calls are coming. No problem. We'll finish this. Then we'll continue. Okay. So I was talking that if it is adsorbed, okay, if it's adsorbed, then it will show negative result again Paul Brunel. But if it is not adsorbed, so positive test here. Okay, in the repetition one. Okay. And second, we take antibodies after serum injection. First is the normal serum. Then we take antibodies after serum injection. Then we take IMN condition, right? So if it is antibodies against serum injection, first, it is adsorbed on guinea pig. It is not adsorbed on your ox RBC. So both will give negative one because both are adsorbed. So both will give negative Paul Brunel test. Okay. On IMN, it is adsorbed on guinea pig. Okay, it is adsorbed on guinea pig. Sorry, not adsorbed on guinea pig. It is adsorbed on good ox. So guinea pig will give positive and ox will give negative. Right. So normal serum, GO. Okay, just remember like this normal serum is a normal serum. Okay, your repetition one results I'm writing GNP will give negative because as of ox RBC will give positive. If it is after serum injections, if it is after serum injections, then both will give negative. Both will give negative. And if it is IMN condition, then it is just the reverse of the upper one. Okay, GNP will give positive, ox will give negative. This is the results. This is the result of your diagnosis. We can have something called the monospot test. It's a modified heterophile agglutination test, a slide agglutination test involving horse RBC. Okay, they're priorly treated and specificity is slightly more. Heterophile antibodies appear early and disappear in 6 months. First week and third week. Okay, appears in first week in about 40% cases, third week in 80 to 90% of cases. But these are not detectable in kids, elderly, and patients with atypical symptoms. You can do nucleic acid hybridization test, the most sensitive test, most sensitive with nucleic acid hybridization. Peripheral blood, you can see atypical TC cells or downy cells. They are downy cells. So enlarged cells, big cells, expanded nucleus, big cells, higher nucleocytoplasmic ratio, abundant vacuolated cytoplasm, TC cells basically, and resembles lymphocytic leukemia. It is a condition which is a very good differential diagnosis of lymphocytic leukemia. Nucleus is basophilic and margins appear indented. Margins basically appear indented by the neighboring cells. Okay, this is a type of, you can see indented margin in this diagram. Very appreciate this type of diagram. Okay. Treatment is IMN supportive measures. Acyclovir for oral hairy leukopenia. Rituximab is a monoclonal antibody to CD20. Okay. CD20. Yes, you bind CD20, right? Okay. So this is the thing. So if I am just repeating, specimen saliva, peripheral blood, inoculation culture is difficult. Detection of EBV specific EBNA is there. EBNA will be detected by EBNA will be detected, right? ELISA, IIF, CFT, then gel diffusion, you can do anything. Heterophile agglutination test is done by pulmonary test. Then you have to confirm it by agglutination adsorption test. You can do monospot test. It's a type of modified heterophile agglutination, slide agglutination. Nucleic acid hybridization is the most sensitive. Peripheral blood smear examination will show atypical TC cells, which are called as downy cells. Okay, downy cells characteristics you have to remember. Treatment is supportive. Basically, acyclovir may be given oral hairy leukopenia and rituximab is a monoclonal antibodies may be effective in this, right? So after this, we'll move to the next virus. Okay. Okay, we are done with HHV4, right? And that is the Epstein-Barr virus. And now we are into the next virus of herpes viridae family. That is the cytomegalovirus. Right, so it is the HSV5, cytomegalovirus. So it is the largest herpes virus. Megalo means big, right? So it is the biggest one. Causes enlargement of cells. Okay, and It is a DNA virus, right? Most common virus for congenital malformations in the West. Okay, it is the most common virus for congenital malformation. Understood? Only one serotype, humans are the only host, also known as salivary gland virus. Okay, basically the virus has the involvement of salivary gland. Okay, so it's a salivary gland virus. I will see the involvement a bit later. In vitro, it multiplies only in human fibroblast cell line. It multiplies in human fibroblast cell line and it produces a characteristic appearance which is the owl's eye appearance. This is the owl's eye appearance. You can look beautiful eyes like an owl, right? It's the owl's eye appearance. Okay, let's come to transmission. Talking about transmission, it is transmitted by kissing, close person to person contact. Okay, trans 2 means transplacentally and transplantation of organ both. Okay, understood. Exposure to maternal body fluids, breastfeeding, contact with saliva or infected people, sexual intercourse. Basically, all the things involving some kind of secretions, kissing, close person to person contact, transplacentally, okay, via the placenta, or transplantation of organs and exposure to maternal body fluids. Okay, exposure to maternal body fluids during delivery and breastfeeding also and contact with saliva or urine of infected people, okay, like young kids and sexual intercourse. CMV replicates here. Okay, main one is the salivary gland, so it's called the salivary gland virus. SRK plus C you can remember. So salivary gland, respiratory tract, kidney and cervix. Salivary gland, respiratory tract, kidney and cervix. Virus is said in cervical secretions, okay, urine, saliva, breast milk, semen, tears. Okay, these are all the secretions, and CMV lies in a latent stage in the salivary gland. It can persist for years. That's why it's called the salivary gland virus. Okay. The virus remains associated with lymphocytes and PMN cells. Clinical features. Most of these infections are asymptomatic. Just remember the thumb rule that majority of viruses are there, majority of pathogens, in fact, produce some kind of asymptomatic infections because our immunity is very good, right? But it can lead to complications like prolonged latency in monocytes and occasional reactivation. It reactivates. If it reactivates in cervical cells, it can cause infection of newborn. Okay, infection of newborn and congenital infections in the newborn is called the cytomegalic inclusion disease. Okay, CID. So the CID of that CID which you watch on television, but it's a CID congenital or cytomegalic inclusion disease. Okay, so you can remember like Mahendra Singh Dhoni is in CID and making a poor joke, right? So MSD, okay, poor joke, कोई हंसा नहीं, HSL, right? So MSD means microcephaly, seizures and deafness, microcephaly, seizures and deafness, and poor joke means purpura and jaundice, PJ, and Hasani means hepatosplenomegaly, HSL, right? So congenital CID, who is there? Mahendra Singh Dhoni, and he made a poor joke and koi Hasani, right? You can remember like this. Okay. Okay. Then again, a characteristic lesion is blueberry muffin lesion. Okay, have you eaten blueberry muffin? It's a tasty one. Basically, in the skin, blackish to blue lesions are there. Purpuric lesions, basically, due to thrombocytopenia occurs, and blueberry muffin lesions are seen in this disorder. Also seen in rubella infection. So they can ask you if you tell that the clinical features of HHV5. To tell that congenital malformations of obviously will tell that most of the are asymptomatic. It can cause congenital cytomegalic inclusion disease, CID. CID means MST. MST means microcephalus seizures and deafness. Right? Yeah, microcephalus seizures and deafness. And PJH means what? Purpura jaundice and HSM. After that, you will say blueberry muffin. They can ask you in which other virus they can cause rubella. Rubella infection may show blueberry muffin disease, right? So RNA virus, rubella will read. So these all points not important for time being. CMV. The risk of congenital infection is maximum if it is occurs early in pregnancy or first time in mother. Okay, first time in mother means there is no immunity, right? And early in pregnancy also in first trimester. Remember, in the first trimester, the infection was causing a stillborn, and the later stages, then hydrops fetalis was being caused, right? Can you remember that? Right. One third of primarily infected mothers, first time infected, transmit the virus to the fetus. Okay. Postnatal infections, so there will be a IMN like syndrome. IMN-like syndrome in postnatal infection. IMN-like syndrome means a heterophile negative IMN-like syndrome occurs in adults and older kids. Okay, again the same things. But there is no exudative pharyngitis and no cervical lymphadenopathy. In IMN-like syndrome, remember fever, lethargy, and all such things we learn, right? CMV is the leading cause of mental retardation in the US. Okay, it's the leading cause of mental retardation. Infected infant may continue to secrete the virus in the urine for years. Congenitally, it can cause retinitis and cerebral calcification. Chorioretinitis and cerebral calcification. Okay, so it resembles congenital toxoplasma. So, chorioretinitis and cerebral calcification are the characteristic features of CMV infection. Okay, look at this. Consume a microcephalic is seizures, some deafness, sensory neural hearing loss. So MSD is done, right? And it is the post joke. Okay, PJ. So periventricular intracranial calcification. Calcifications are there. Chorioretinitis is there. I told you earlier. Right? Chorioretinitis is cerebral calcifications. Okay. So MSD, PJ. PJ means that purpura. Purpura, which was causing a blueberry muffin rash, right? Blueberry muffin lesions or blueberry muffin is also seen in rubella. Remember? Yes. That was there, and Hastani means hepatosplenomegaly. Let's see hepatomegaly, hepatosplenomegaly. CMV can cause disease in immunocompromised. In AIDS patient, organ transplant recipients, it can cause disease, right? So in AIDS patients, ADR, just remember ADR, it can cause diarrhea and retinitis in AIDS recipient, in the AIDS patients, right? Seen when CD4 count is less than 50. This you have to remember, right? Less than 50. Pneumonia is there. Then organ transplantation, most common viral pathogen infecting organ transplant recipients. The most common viral pathogen which is infecting organ transplant recipients. Okay. CMV includes both intranuclear and intracytoplasmic inclusion body. Okay, it includes both intranuclear as well as intracytoplasmic inclusion body. Just a minute. Understood, this much is there. CMV can cause disease immunocompromised, 8% ADR, you'll remember, diarrhea and retinitis. Organ transplantation, you'll see it's the most common viral pathogen which is causing an disease in organ transplantation people, pneumonitis, esophagitis, and hepatitis. Okay, lab diagnosis again. You can do viral isolation in the human fibroblast cell line because it multiplies in human fibroblast cell line, produces owl's eye appearance, right? Microscope will see inclusion body and owl's eye appearance. Antigen detection, PP65, important. PP65 is present nucleocapsid with 2 nucleotides. Antibody detection by ELISA, IIM, IHM, and CT. Okay. Treatment, ganciclovir. Ganciclovir is using at CMV retinitis. In CMV retinitis, what happens? We give an intraocular injections, right? Intraocular injections of that ganciclovir, right? And pneumonia. Then valganciclovir, it is also used for CMV retinitis. For pneumonia, you can give valganciclovir. Pseudofovir, you can give for CMV retinitis, and fomepizole is also there. Ganciclovir, valganciclovir, pseudofovir, and fomepizole, right? So no vaccine is available. So we are done with the 5 herpes viruses, and now let's move on to the next virus. Okay, so the next virus we have is HHV6. Okay, HHV6 is the human herpes virus 6. It infects T cells. Okay, infects T cells. Which was the virus infecting B cells? Just come on, come on. Yes, Epstein-Barr virus. This virus, CD6, is infects T cells. Okay, T cells, and you can just remember by this receptor 46. Just can you hear the sound of T? Yes, T. So T cells and CD46 is the receptor. So and 6 is the herpes virus, right? HHV6. Understood. So this is the fact. Human herpes virus infects T cells, CD46 receptor. Two variants, 6A and 6B are two variants. Transmission by infected oral secretions, right? In kids, it calls sixth disease, or exanthem subcatum. Okay, exanthem subitum. Okay, and it is also called as roseola infantum. By 6B, it causes IMN-like syndrome in older people. Done. So HHV 6 is done. Now we'll move to the HHV 7. Understood? Okay. Let's start HHV 7. Right. So HHV 6 we saw what it was causing sixth disease, or exanthem subitum, or it's called as the roseola infantum. Okay. Can you recall a virus which was causing the fifth disease? Can you recall a virus which was causing the fifth disease? And what is the name of the fifth disease? What's the name? Can you recall? Yes. If you have recalled, it's well and good because we learned that the fifth disease was being caused by, sorry, what I'm writing. Okay, fifth disease was being caused by parvovirus, right? Parvovirus was causing the fifth disease, and the name was erythema, erythema influenza. Okay, influenza. So it is the fifth disease. Let's move on to HSV7. HSV7 is a human herpes virus 7. Again, the same as herpes virus 6. Okay, it also infects the T cells, transmitted by infected oral secretions. Also the same. Okay, this was also transmitted by infected oral secretions, and this homology is said with HSV7, associated with fever, seizures, respiratory symptoms, and pityriasis rosea. This line is last line is important. It is associated with pityriasis rosea. Okay, rose like spot. Okay, and pityriasis is a rose like spot. Okay. Next we have the HHV8, which is the Kaposi sarcoma virus. Okay, KSHV means Kaposi sarcoma herpes virus. The Kaposi sarcoma associated herpes virus or Kaposi sarcoma herpes virus was first discovered in 1991 in a patient of Kaposi sarcoma. Belongs to gamma subfamily of herpes viruses. Okay, gamma subfamily, and infects the B cells. It infects the B cells. So it belongs to gamma subfamily of herpes virus, infects the B cells, and increased prevalence in areas of Africa. Okay, because transmitted oral secretions, and decreased prevalent areas are North America, Asia, Northern Europe. Okay, affects adults and most commonly transmitted with the sexual route, common in homosexual males. Okay, infects adults, sexual route, and homosexual males are most commonly affected, right? Then less common modes of transmission are organ transplantation, blood transplantation, and injection drug abuse. Okay, by organ transplantation, blood transplantation, and drug abuse, it is less commonly transmitted. Okay, Kaposi sarcoma is a AIDS defining malignancy. It is a AIDS defining malignancy, right? So soft tissue sarcoma, vascular origin. The soft tissue sarcoma, soft tissue infection of vascular origin, it's a AIDS defining illness. If Kaposi sarcoma is present, then definitely you should check for HIV, right? So it's red to purple colored growth under the skin. So red or purple colored growth, skin or oral or oral mucosa also, mouth and oral mucosa are affected also. This question, this image was given in NEET PG. Okay, this is a NEET PG image, and Kaposi sarcoma of oral mucosa is there. Also, it can cause primary effusion lymphoma, Castleman disease, and fever and rash in immunocompromised people. It can cause primary effusion lymphoma, Castleman disease, and fever and rash. So you learned what is Kaposi sarcoma. Kaposi sarcoma lesions are red or purple, red to purple colored lesions. Okay, they may be intermediate or fully red or fully purple. It may be there, and this occurs under the skin. Oral and mouth may be affected. Oral mucosa or mouth may be affected. This is the image. Also, it can cause primary effusion lymphoma, Castleman disease, and fever and rash in immunocompromised. Right. So I will suggest you to make a list on the disorders, the infections which are having a fever plus rash presentation, because this fever plus rash in many times clinical practice serves a important differential diagnosis of many diseases. So you should have a gross mindset that you should read like this, that which infections can cause this type of disorder, right? Like if I ask which infections can cause primary effusion lymphoma, there are many. Okay, like infections can present with fever plus rash. So, dengue virus can be a differential diagnosis, right? So you have to know this in total, right? Okay. After finishing all the herpes viruses, we are with another virus, which is the rubella virus. Okay, rubella virus. Okay, let's start with the rubella virus now. So, should we start with rubella virus? Yes. Okay. So starting with rubella virus is an RNA virus, positive strand ssRNA virus like HIV. Okay, it is also type of RNA virus. Family is Togaviridae. Only virus of Togaviridae family which is not arthropod borne. It's not the arthropod borne virus. So when we read about arthropod borne viruses like dengue virus, chikungunya virus, and all, we'll see. Okay, it is not a arthropod borne virus, but belongs to Togaviridae. Okay. Transmitted by elimination of respiratory droplets. Again, one antigenic type is there, enveloped and icosahedral symmetry. Only and the natural hosts are humans, and disease occurs worldwide. What are the diseases? Rubella, German measles, or 3-day measles. Rubella, German measles, or 3-day measles. So rash lasts for 3 days only. Okay. Incubation period is 14 to 21 days, like your varicella virus, varicella zoster. Okay, but followed by brief, same thing will occur. Brief prodrome of fever and malaise will occur, followed by macular-papular rash. Macular-papular rash starts on face and progresses downwards. Okay, but the varicella zoster starts on trunk, then progresses to head, neck, and all the extremities. Right. Posterior auricular lymphadenopathy is a characteristic feature. Just stay clear of this history. This posterior auricular lymphadenopathy is a very characteristic finding which you should know. Okay, this is because of antibody antigen mediated vasculitis. Okay, one infection leads to lifelong immunity. Polyarthritis in adult females, deposition in joints. Okay, polyarthritis and deposition in joints will occur. Rubella virus is a teratogenic virus. Rubella virus is a teratogenic virus. When a non-immune pregnant lady gets infected in the first trimester, virus can cross placenta even. Okay, this virus can cross placenta and cause congenital rubella syndrome. Congenital rubella syndrome. Okay, another thing, congenital. Can you recall? Just a thing which I told you. Congenital one. CID. Recall CID. MST post joke. कोई नहीं आता. Yes. Virus can cross placenta, can cause congenital rubella syndrome. Congenital rubella syndrome is classified by the heart, eye, and the ear. Okay, the heart, eye, and ear diseases. So, in heart disease, the most common is the patent ductus arteriosus. The ductus arteriosus, the embryological name, this basically remains patent. Okay, patent ductus arteriosus, and it persists. Okay, it doesn't regresses to the ligamentum arteriosum. It persists as a ductus arteriosus. Ductus arteriosus is basically the hole, right? So a ligament type of structure is formed in the adult stage, but here it doesn't happen. So heart defects most common is the patent ductus arteriosus, right? Ocular, the most common one is the salt and pepper retinopathy. Okay, salt pepper retinopathy. Salt pepper retinopathy. You can see the type of salt and pepper. This type of dotted appearance occurs, and the second most common is the cataract, right? So heart defects, patent ductus arteriosus. Others are the pulmonary artery stenosis, ventricular septal defect, VSD can occur. Okay, this type. Ear defects. Ear defect is the sensory neural hearing loss. The sensory neural hearing loss. Okay. Also, most common defect in rubella. Most common defect in rubella is sensory neural hearing loss. Some of the infected children in utero can continue to excrete the virus for months after they are born. So, it is a significant public health hazard. It is a significant public health hazard. Some of these congenital status are asymptomatic without any congenital malformation that can be detected by viral isolation and increased antibody titers. Okay, some of the congenital status are asymptomatic. So you just know now that rubella virus when you start the common features, you know that are transmitted by this antigenic types and all. Toga viridae is this statement is important. Only virus of toga viridae, which is not arthropod borne. Rest of the virus is arthropod borne, right? And about the disease, the rubella, the measles, the rubella, German measles or the 3-day measles, right? This is the measles lasts for 3 days, right? And question period 14 to 21 days. This is important. Brief prodrome of fever malaise occurs. Then after maculopapular rash, which starts from the face itself. This maculopapular rash starts from the face itself, right? And posterior auricular lymphadenopathy is a characteristic feature. Polyarthritis may occur, right? And rubella virus is teratogenic. It can cause cross placenta and cause congenital rubella syndrome also. Congenital rubella syndrome is CRS. Okay, if you get the abbreviation that CRS, you don't get confused. Congenital rubella syndrome. I told you that heart is involved, eye is involved, and ears are involved, right? So, in heart, your most common one is the patent ductus arteriosus. You can have your ventricular septal defect also, right? And pulmonary stenosis, PAS. PAS and septal defect can also be there. And ocular diseases, the most common one is the salt pepper retinopathy. And the second most common is the cataract, right? And ear defect, you can have sensory neural hearing loss, which is the most common in rubella. And the infected children, they continue to excrete the virus for months and months after they're born. And most of them are, like some of them are asymptomatic, not most. Okay, this much is important for rubella point of view. Lab diagnosis of rubella. Okay, what are the lab diagnosis part? So culture. Culture is the part which is important here. Rubella virus itself doesn't produce any cytopathic effect. Doesn't produce any cytopathic effect. It usually interferes with the cytopathic effect by ecovirus. It interferes. It is a viral interference. Remember, I told you about the cytopathic effects of the chimti. Remember chimti, the side of the that affects the detection of virus. How can it know that a virus infection has occurred? Okay, so you had the cytopathic effect, heme adsorption, viral interference, metabolic invasion, and other things, right? So you just remember this chimti, and it usually interferes with cytopathic effect by ecovirus. So it interferes with cytopathic effect by ecovirus, right? And method. What is the method? Add patient's sample to the cell line. Add the sample of the patient to the cell line. Rubella virus will infect the live cells. Superinfect the culture with ecovirus. If there is no cytopathic effect by ecovirus, basically what is happening that rubella virus is inhibiting the cytopathic effect by ecovirus. Okay, so basically what we are doing, we are adding the patient sample. We are infecting it with rubella. So now we have what we have. We have rubella infected sample. Now we have to super infect. Okay, we have to super infect. I'm writing SI. Super infect with what virus? Ecovirus. Okay. If ecovirus is producing the cytopathic effect, if ecovirus is producing a cytopathic effect, that means rubella is absent because rubella is interfering with ecovirus. Na? Okay, but if rubella is absent and ecovirus will produce a cytopathic effect, then rubella is absent, right? So there is no cytopathic effect by ecovirus. It indicates that rubella is present in the sample, and rubella is present means it is a positive test, right? And this whole phenomenon is called the viral interference, right? It's done in the ecovirus and the rubella virus. Serology with a fourfold increase in antibody titers. You can see convalescent and acute sera sample. ELISA and heme adsorption can be done. RT-PCR can be done. Treatment is supportive. No antiviral is specified here. Done. So that was our discussion on rubella virus. Okay. Now let's move on to the next virus. Okay, guys. So let's start with the polio virus. Polio virus is an important virus. So do read it very carefully. Belong to the picornaviridae family. Okay. Three antigen type immunities, type specific immunity we have. So type 1, type 2, and type 3. So we have three types of polio viruses. So type 1 is known as the Brunhilde Mahoney strains. Type 2 is Lansing and Mef1 strains, and type 3 is Leon socket strains. Okay. So if you just read the names many times, then you can retain it. Otherwise, it's difficult, right? So Brunhilde Mahogany is the type 1. Brunhilde Mahogany. Type 2 is Lansing and F1, and type 3 is Leon and socket. Okay. Okay. So type 1 is the most common serotype responsible for endemics of poliomyelitis, and type 2 is Lansing and MEF2, that is the most antigenic, okay, most antigenic and easiest to be eradicated. And Leon socket stain haven't been seen till since 2013, right? So polioviruses, two antigens are there: capsid antigen and dense antigen, C antigen and D antigen, capsid and dense. So C is associated with non-infectious virus, dense is associated with whole virion. Capsid is less specific, dense is type specific. Anti-C antibodies are not protective, anti-D antibodies are protective. So you can see the positive points are there, yeah, right? It associated to whole virion type specific, and anti-D antibodies are protective. And C is associated non-infectious, okay? It's less specific, and anti-C antibodies are not protective, okay? And potency of injectable and field vaccines measured in terms of D antigen units, DAU. DAU is the D antigen units, which measures the potency of injectable and field vaccines, right? Pathogenesis and clinical features. So incubation period is 1 to 2 weeks or 7 to 14 days. 7 to 14 days is the incubation period. Poliovirus is transmitted by fecal-oral route. Is the most common route. Most common route of transmission of poliovirus is fecal-oral route, right? And followed by respiratory droplet inhalation, very rarely due to your conjunctival inoculation, right? So pathogenesis 1 to 2 weeks or 7 to 14 days, the incubation period. transmitted by fecal oral route, okay, and followed by respiratory droplet inhalation or very rarely by conjunctival inoculation. Virus multiplies locally in the gut, okay, in the intestinal epithelial cells. It multiplies and also in some lymphoid tissues like tonsils and Pierce patches. Viruses use CD155 receptor, very important. It uses CD155 receptor to enter the host cell. Virus multiplies in the regional lymph nodes. Okay, it multiplies in the regional lymph nodes and enters the blood and causes primary viremia. Okay, after entering the blood, it reaches the reticuloendothelial system and again enters the blood. Okay, the reticuloendothelial system again enters the blood. Okay, it's a secondary viremia and it changes its route, causing hematogenous route, reaches the brain and spinal cord. Okay, so basically what is happening is that basically we are fecal-oral route is the most common method of transmission, right? By respiratory droplets too, and by very rarely by conjunctival inoculation too in some specific procedures. So viruses is basically multiplying in the gut, right? Gut, and it is using the CD155 receptor. After multiplying in the gut, it is going to the lymph nodes, so multiplying the regional lymph nodes. The regional lymph nodes will drain the virus, send the virus to the blood. Okay, if it comes to the blood first time, then primary viremia. After primary viremia, it goes to the reticuloendothelial system, multiplies there. After that, it is causing a secondary form of viremia, secondary viremia. Then after it reaches brain and spinal cord. And during tonsillectomy, this virus has a neural spread. You must be knowing this fact, and the virus can spread through the nerve, right? So during tonsillectomy, virus may have spread by glossopharyngellum in the tonsillar fossa. So eventually, virus targets the motor nerve endings, and cause muscle weakness and flaccid paralysis. Important point: it is a flaccid paralysis. Okay, poliomyelitis is a flaccid paralysis. Okay. Degeneration of neurons, earliest change is a degeneration of Nissel's body. Nissel's body, the RER and the ribosomes are present in the nerve, right? So Nissel's bodies. So earliest change in the poliomyelitis is degeneration of Nissel's bodies, right? Clinical features 90 to 96% patients are asymptomatic. Okay. 5% abortive infection, that is fever, malaise, sore throat, headache, all these are happening. 1% present with aseptic meningitis, influenza like illness, and non paralytic poliomyelitis. Okay. So 5% abortive infection, 1% aseptic meningitis, and causing non paralytic poliomyelitis. So less than 1% develop paralytic poliomyelitis. So we have a general intuition that if poliovirus is infected, then the person will develop poliomyelitis and develop paralysis. The person will limp and walk. So and so we have a pre-concept in our head which is incorrect. So 90 to 96% patients nearly are asymptomatic. Okay. 5% patients are abortive infection, like very mild infection. 1% have meningitis, but less than 1% has, paralytic poliomyelitis. So it is a flaccid paralysis, right? Proximal muscles are involved earlier than distal muscles. Proximally, the proximal muscles are more involved. The brainstem involvement is brainstem is involved. Okay, the brainstem is involved, then there will be a respiratory failure. You can say the diaphragmatic muscles are also involved. If these are involved, then also respiratory failure can happen, right? Occasionally, cranial nerves are involved. Occasionally, cranial nerves are involved. Okay, no sensory losses there. Lab diagnosis. So, specimen collection, transport, culture, antibody detection, molecular methods. These things are there. Then we'll wind up. Specimen. Up to 1 week of illness, the virus may be recovered from the throat swab. So basically, it is going to the oral cavity first, right? Fecal route we are taking. So up to 1 week. Up to 1 week, you can recover it from throat swab. Okay. Up to 6 to 8 weeks. Just see the incubation period. 1 to 2 weeks is the incubation period. Okay. So if 1 to 2 weeks in the incubation period, up to 1 week, the virus may be recovered from throat swab because it has not produced any symptoms till date, right? And rectal swab and feces has may be isolated up to 6 to 8 weeks. Okay, 6 to 8 weeks. Long-term excretion of virus occurs in immunocompromised. Very rarely, virus can be isolated from blood and CSF also because viremia is occurring, right? So sometimes the blood may be infected with viruses. It may show viremia and CSF2 because it is in 1% patient it does cause aseptic meningitis, right? In 5% it is not abortive infections. 90 to 96% are asymptomatic. Less than 1% present with flaccid paralysis. Sewage testing is done for epidemiological purposes. Transport frozen specimens are to be transported. Culture is the primary monkey kidney cell line. Okay, primary monkey kidney cell line. Virus can be detected by CPE antigen detection and PCR. CPE antigen detection and PCR. Okay, these are the ways in which the virus can be detected. So CP cytopathic synchronization and degeneration of cells will occur. Antigen detection by IIF and neutralization test, which is poliovirus specific antisera. Poliovirus specific antisera is used in the antigen detection, and PCR to detect the BP1G. BP1G is detected by PCR. Okay, you can remember poliovirus, right? Poliovirus PB. You can reverse it BP1. So BP1 gene can be detected. So these are the lab diagnosis of poliovirus. Antibody detection, fourfold rise in antibody titer, neutralization test, and complement fixation test you can determine. So first infection with poliovirus lead to antigenic type of serotype specific antibody formation. To that serotype the antibody is formed. Okay, and if subsequent infection occurs, then antibodies against the group specific antigen is produced. Okay. Molecular methods, real-time multiplex reverse transcriptase PCR. Okay, real-time PCR, which is a form of multiplex PCR only. This is used. Reservoir of poliovirus is humans, only humans. Most cases are subclinical. No chronic carriers are there. Sources of infection are feces and oropharyngeal secretions. Age, young kids and infants are more prone to infection in developing countries, but in developed countries, older kids are more prone. Patients are infectious from 7 to 10 days before onset to 3 to 4 months. Okay, up to 3 to 4 months. We tell the 2 to 3 weeks thereafter is most accepted one. 3 to 4 months virus may be present, right? Okay, so that finishes our poliovirus. Let's move on to the next. Okay, so now we are into a fresh virus, and this will be the last virus we will discuss since it is almost 1:24 a.m. and I'm recording this lecture. So I guess you all may be bored and I'm sleepy. So we'll continue the next part of the virology, like the AIDS virus, coronavirus, and other that influenza virus, hepatitis virus in another session, right? So virology will be two sessions: virology one, virology two. Rabies virus. Okay, what is rabies virus? So basically, what is happening that it is a RNA virus. Family is a Rhabdoviridae. Rhabdoviridae family. Two genera are there: Lyssa virus and vesiculovirus. Okay, Rhabdoviridae, Lyssa virus and vesiculovirus. If Lyssa virus contains rabies and rabies-related virus, which is vesiculovirus causes vesicular stomatitis virus. Vesicular stomatitis virus. So two genera, Lyssa and vesiculo. Lyssa will be a rabies virus and rabies-related, and vesiculo will be vesicular stomatitis virus. Right. Okay, this genus Lyssa virus causes rabies. Rabies is a rapidly progressive acute infectious disease of CNS of both humans and animals. Okay, it is a disease of both humans as well as animals, transmitted from other rabid animals to humans. Right, human to human transmission is also theoretically possible, but not practically seen. One serotype, seven antigenic variations. Structure is bullet-shaped virus. You can see the structure here. It's a bullet-shaped virus. Okay. So on the top, we have the glycoprotein 6, which is antigenic. It's the antigenic glycoprotein 6. A lipid bilayer is there. So lipid bilayer is there. L and P proteins are there, which are RNA polymerases basically. L and P proteins, you can see. There's red ones and blue ones. A matrix protein is there, and helical nucleocapsid. Okay, RNA and N protein. Glycoprotein G, which I told here, this antigenic one, glycoprotein D, is basically important for pathogenesis and neuroinvasion, right? Animal susceptibility can affects warm-blooded animals, including humans. Okay, it affects warm-blooded animals. Let's see. So, which animals? Some group of animals are low susceptible, some are moderately susceptible, and some are highly susceptible. Okay, low. O sound is there, right? So opossum is low susceptibility. Moderate susceptible, like dog, horse, sheep, goat. Okay, all the you can say domestic animals, right? Dog, horse, sheep, goat, this type of things. Monkey, non-human primates, right? Highly susceptible. Highly susceptible means some animals which are discrete animals. You would say bat, raccoon, hamster, cattle, cat, rabbit. Okay, just not closely related. Very highly susceptible. Very highly susceptible. You can remember wild animals like fox, jackal, wolves. Okay, just remember this. Cotton rats also. Can you remember this? Just you just know it grossly. It's not mandatory to remember this so well. But if you just read it 2 3 times, then you can retain it, right? So this animal susceptibility point you have to write. Rabies virus have two variants. Okay, one is street virus, one is a fixed virus. Okay, let me make you understand what is street virus, what is fixed virus. Street virus is anything which is freshly isolated strains in the lab. Okay, so freshly isolated ones, and it mimics the wild virus. So we have a wild virus here, and this wild virus has two variants: wild virus and street virus and a fixed virus. Okay, so basically, when we isolate the virus first in the lab, then we make a strain the street virus. The street virus is exactly similar to that of wild virus, right? So it has a long variable incubation period and intracytoplasmic inclusion bodies are formed. This is a street virus. Okay, street C and S. You can just similar sounding intracytoplasmic is T S sound. Okay, street virus. Fixed virus. If the fixed virus is propagated in rabbit, okay, a brain to brain, a serial brain to brain passage is done. A serial brain to brain passage is done. Okay, the street virus is propagated in rabbit, then it changes to the fixed virus. Okay, so if there is a serial brain to brain passage, the street virus changes into a fixed virus, right? Okay. This fixed virus does not produce intracytoplasmic inclusion bodies, does not multiply in extraneural tissues, does not infect salivary glands. Okay. This does not produce intracytoplasmic inclusion bodies. This produced by the street virus. It does not multiply in extraneural tissues. It does not infect the salivary glands even. Okay, but it multiplies rapidly in neural tissues. Okay, this is more kind of neural type. The brain thing is there, fixed thing is there, right? So incubation period of this is 4 to 6 days. Pathogenesis. What is the pathogenesis of rabies? So virus is transmitted to humans by the bite of a rabid animal, right? So in India, dog bite cases are more common. Dog bite cases are very common, right? Dog bite 97%, cat bite 2%, and cat scratches basically. This can be considered as mongoose and jackal less than 1%. Okay, less than 1%. And bat bite. Bat bite is more common in West, right? Bat bite also can transmit rabies here, okay? Which is they're like more common in the West, not in India. Rabies due to human bite is theoretically possible but not reported. I told you earlier. Non-bite exposure. If a or animal is not biting, can it transmit rabies? Yes. Licks abrasion on mucous membrane. If the animal is licking the abrasion on the membrane or on your skin, there is a wound and the animal is licking the wound. Animals tend to lick the wound, right? If a rabid animal tends to lick the wound, then rabbit virus definitely go inside, right? Inhalation of aerosols containing the virus is very rare. Corneal transplantation can occur, but very rare. Spread of virus. How the virus spreads? Virus multiply locally in the muscles and connective tissue. Muscles and connective tissue locally. Then entry of virus into peripheral nerves. Okay. After multiplying the muscles and connective tissue, it moves to the peripheral nerves, like peripheral nerves. It binds to the nicotinic ACH receptor. Basically, this virus is a kind of neural virus. So it is binding on the NMJ, neuromuscular junction. Right? Neuromuscular junction. So basically, virus binds to the nicotinic ACH receptor on the neuromuscular junction. Then it spreads neuronally, neuronal spread. Virus spreads centripetally along the peripheral nerves by retrograde fast axonal transport. Retrograde fast axonal transport. The viruses spread centripetally along the peripheral nerve. Reaches the dorsal root ganglia and ascends up to the CNS. It reaches the dorsal root ganglia and ascends up. Okay. So basically, virus is multiplying the muscles and connective tissue first. Okay, it's multiplying there. Then it's entering the peripheral nerves. Okay, trying to enter the peripheral nerves and spreading neuronally. Neuronal spread is occurring by retrograde transport. A retrograde fast axonal transport and reaches the dorsal root ganglion and reaches up to CNS. In CNS, virus rapidly disseminates to hippocampus and cerebellum. Okay, disseminates to hippocampus and cerebellum, and then virus spread along the sensory and autonomic nerve to salivary gland, pancreas, kidney, retina, cornea. Okay, these structures. So virus is disseminating to hippocampus and cerebellum and spreading via sensory and autonomic nerves to this salivary gland, pancreas, kidney, retina, cornea. There is no viremia. Okay, there is no. You can see there is no mention of any bloodstream. Okay, there is no viremia. So this is the whole picture. You can take a screenshot if you want. Virus is said in saliva of a rabid animal. Histopathological changes in brain parenchyma. This is an important. So you can remember MPBN. MPBN, what is MPBN? Mononuclear cell infiltrate. Perivascular cuffing. Perivascular cuffing means the blood vessel is there, right? So the lymphocytes will be arranged around it. You can see in the diagram also. The perivascular cuffing is there. Babes nodules. Babes nodules is present in glial cells. You can see these are the Babes nodules which are present. Babes nodules and Negri bodies. Negri bodies is very important. Negri bodies is a intracytoplasmic inclusion body. Negri body is a intracytoplasmic inclusion body which is seen in the case of rabies. Right? Detection confirms the post-mortem diagnosis of rabies. Okay. So you can't get Negri bodies in an alive patient. How will you get? Will you get it's a post-mortem feature, right? You can't get it in a alive patient. Negri bodies are seen Purkinje cells, pyramidal cells, cerebellum, hippocampus. Purkinje, pyramidal, cerebellum, hippocampus. Hippocampus is the most common site of Negri bodies, right? This much things you have to remember. So, what are the histopath features? Mononuclear cell infiltrate. Okay, mononuclear cells infiltrate the inflammatory cells, basically inflammatory condition, right? And perivascular cuffing, Babes nodule, Negri body. Prolonged incubation period 20 to 19 days, 1 week to 19 days. Okay, basically on average you have 20 to 90 days, 20 to 90 days. Okay, but it can be extend up to 20 years even. Okay, 19 to 20 years. It directly depends on the distance the virus has to travel to the bite to the CNS. Okay, it depends on the distance. Decrease incubation period. How the incubation period will be decreased? If the bite is nearer to the CNS, bites on head and neck. If the bite is nearer to the CNS means upper limb bite will cause. So upper limb, kids also because they have shorter height, right? Kids, short people, and other things like laceration, genetic predisposition, decreased immunity, these all can cause a decrease, less incubation period. Higher inoculum, increased virulence. If the virulence is more, okay, if there's a virulent strain, then it will take less time to infect. It will infect you straight away, right, and cause destructive features. Okay. Then we have some of the okay. Then we have some of the clinical features of rabies. Okay, let's pay attention to the clinical features. So we have first a prodromal phase. Okay, a prodromal phase. Prodromal phase lasts from 2 to 10 days. 2 to 10 days. Fever, anorexia, nausea, malaise, sore throat. Just normal features, non-specific features. One important one is photophobia and abnormal sensation at the site of bite. Okay, pain, pruritus is all. Photophobia, the person suffers with photophobia and abnormal sensation. This is a prodromal phase, a mild phase. Then we have the second one, that's the acute neurological phase, which can be encephalitic phase or furious rabies in 80%, or paralytic phase or dumb rabies. Okay, so rabies may be a furious one or a dumb one. Interesting. The virus can be a furious one or a dumb one, right? So either it can be encephalitic rabies, a furious one means hyperexcitably. Okay, autonomic disturbances like salivation, lacrimation, all secretions will increase. Okay, because the person will come to the sympathetic state now, the parasympathetic state now. Okay, so we can have a furious form of rabies, or it can be paralytic or dumb rabies. Dumb rabies is in partially vaccinated people. The rabies cannot develop full-fledgedly, right? It sits like a dumb person, like lethargic person. Okay, so in this type of rabies, in your furious rabies or encephalitic rabies, we have priapism, cardiac arrhythmias, and goose flesh. Goose flesh, the goosebumps. You know, goosebumps are the if you put your finger in the water, then this type of striations appear. This is goose flesh. Cardiac arrhythmias. Arrhythmias, you know, the abnormal beating of heart, and priapism is the painful erection of the penis. Okay, and hydrophobia and aerophobia will be there. Okay, this is a very important. Due to brainstem neurons, the person suffer from hydrophobia. The person will fear water. The person will fear air. Okay, he just can't drink water. If you give a glass of water to the person will just act very widely. Okay, the person will just try to like this type of things. Okay, like he will start behaving like a dog. People think that the rabid person basically mm, behaves like a dog. Okay, but it's not the case. Okay, if it's bitten by some monkey also, he will say he will be behaving like a dog only. Okay, but that's not the case. It may resemble, but it's not that if you are bitten by a cow and you suffer from rabies, you will do moo moo. Okay, it's not the thing. Paralytic rabies. Paralytic rabies, the flaccid paralysis will be seen again, like poliomyelitis. Starts in the bitten limb and progresses to the quadriparesis. No, quadriparesis will be there. Then coma and death. Third stage is coma and death. A prodromal phase, acute neurologic phase. Acute neurologic phase may be a dumb rabies. Dumb rabies means the encephalitic phase, or a paralytic or dumb rabies. Okay, sorry, furious rabies is the encephalitic phase, and paralytic or dumb rabies is your dumb phase. Like coma and death, usually death occurs in 14 days. Usually death occurs in 14 days. Okay. Lab diagnosis. Rabies antigen detection. By direct immunofluorescence, you can do rabies antigen detection. Rabies nucleoprotein antigen you can detect. And increase sensitivity specificity is the gold standard test, but you don't require this lab diagnosis, right? The person will, if you are, okay, from microbiology point of view, you can say that lab diagnosis is important, but you have to write in the questions, right? But do you think practically the person you'll be doing a antigen detection in the person coming with rabies? No, right? Because the person will start behaving like you can diagnose it clinically. Okay, you diagnose it clinically. You can just take a history of bite, right? The person will come with a bitten one and start behaving like a rabid person, right? You don't have to do an antigen detection to confirm it. Best specimen is the hair follicle in the nape of the neck. Okay, in the nape of the neck, in this posterior part, the hair follicles, and this hair follicle is the best specimen for detecting the rabies antigen. Rabies antigen, right? It's a gold standard also. Corneal impression smear may be made, not much important. Okay, okay. Viral isolation, inoculation of specimen into mouse. Suckling mouse cell line. Do you remember? I talked about rabies virus also. Okay, in suckling mouse cell line. If you have remembered, well and good. Otherwise, just right. So encephalitis and death will occur. Brain biopsy will taken to look for Negri bodies or rabies antigen. Cell lines: mouse neuroblastoma cell line and baby hamster kidney cell line. Okay, mouse neuroblastoma cell line and baby hamster kidney cell line. It goes in 2 to 4 days. Tested by DIO. Antibody detection in CSF and serum. CSF and serum antibody detection will do. So CSF most significant antibodies appear early only in infected people, but in serum antibodies appear late, right? And they may be present in vaccinated people also. Antigen used is against the glycoprotein G, the outer one. Antibody detection is done by mouse neutralization, not much done. RFFIT is the rapid fluorescence focus inhibition test. This is important. RFFIT is used in rabies, rapid fluorescence focus inhibition test. And FABN test. FABN test is fluorescent antibody virus neutralization test. So RFFIT and FABN test may be done. Viral RNA detection by RT-PCR. RT-PCR detects this nucleoprotein and large structural proteins. Sample maybe brain tissue, saliva, skin biopsy may be there. Okay, then you can detect the Negri bodies. It's a post-mortem diagnosis. It's a cytoplasmic inclusive body, eosinophilic and basophilic inner granules, 2 to 10 millimeter in shape, spherical to oval. Okay, pathognomonic feature of rabies. Pathognomonic feature of rabies. Stains for Negri body. What are the staining techniques for Negri body? HND and cellular. HND and cellular. Okay. For basophilic granules, Manston, MANN, Manston. For basophilic granules, you use Manston. IHC staining, you can use peroxidase labeled antibodies. Okay. Talking about treatment, no supportive treatment. Symptomatic treatment is given. Patient is kept in isolation, sedative and anti-anxiety drugs, hydration and urination are maintained. Okay. So that completes our rabies. Let's recap quickly. Rabies virus, RNA virus, rhabdomyretic, Lyssa and vesiculovirus, vesicular stomatitis, rabies and rabies-related viruses. One serotype, seven antigenic variations. Bullet shaped viruses, the glycoprotein G, which is antigenic. LNP proteins are RNA polymerases. Glycoprotein G is important for neuroinvasiveness and pathogenesis. Animal susceptibility, I told you that low susceptible opossum, moderate susceptible all the domestic animal, dog, cat, horse, sheep, goat. Okay, cat is a highly susceptible one. Okay, just all the domestic animals except the cat. Cat, highly susceptible, rabbit, cat, hamster, raccoon, bat. Okay, all this. Very highly susceptible, you have fox, jackal, okay, and a cotton rat. Rabies virus may be of two variants, street virus and fixed virus. Street virus in the freshly isolated mimics the wild viruses and produces intracytoplasmic inclusion bodies. And this fixed type of virus basically is produced from the serial brain to brain passage of the street virus, right? And the street virus changes to the fixed virus. It doesn't produce any intracytoplasmic inclusion body, doesn't multiply in extraneural tissues, doesn't infect salivary glands, doesn't does all. But it multiplies rapidly in the neural tissues and incubation period is 4 to 6 days. The pathogenesis, it is the bite of a rabid animal. A dog bite is 97%, cat bite 2% and all. Bat bite in West and non-bite exposure like licking, any kind of inhalation of aerosols and corneal transplantation. Spread of the virus and multiple muscles and the connective tissue, then peripheral nerves, it spreads, binds to the nicotinic acetylcholine receptors. Okay, binds to the nicotinic acetylcholine receptors, and after that, it is a neuronal spread, retrograde neuronal fast external transport. This is the dorsal root ganglion, ascend up to the CNS. It ascend moves to the DRG and ascend up to the CNS, and in the CNS it disseminates to the other areas like salivary gland and say hippocampus and cerebellum. In hippocampus, the most common site of production of negri bodies, and virus spreads to the salivary gland, pancreas, kidney, retina. No remains other. Histopathological changes, you can include MPBN. MPBN, I told, MPBN is a mononuclear cell infiltrate, perivascular cuffing of lymphocytes. Babes nodule, negri body. Babes nodule, negri body. Okay. So negri body, I told, intracytoplasmic inclusion. Detection is the postmortem diagnosis. Negri body is a single Purkinje cell, pyramidal cell, cerebellum, and hippocampus. Incubation period is prolonged and variable. 2 to 90 days is the direct infant distance. Okay, decreased incubation period, the more the proximity to the head and neck or the CNS, more is the thing. Short people and kids have shorter incubation period. Lacerations, any kind of genetic predisposition, immunity decrease, higher inoculum, high virulence, all of this. Clinical feature, I told you that we have a prodromal phase, 2 to 10 days is prodromal phase. Just simple symptoms, fever, anorexia, malaise, all this. Okay, photophobia and abnormal sensations may occur. Acute neurologic phase, we have encephalitic rabies, the furious rabies, and a paralytic rabies, which is the dumb rabies. Encephalitic rabies and paralytic rabies. In encephalitic rabies, you can have hyperexcitability, lucid intervals, autonomic disturbances, and in paralytic rabies, we can have a partially infected persons, right? And encephalitic rabies, we may have some kind of priapism, okay? The painful urination, cardiac arrhythmia, and goose plus. Priapism, cardiac arrhythmia, goose plus hydrophobia, plus aerophobia, right? Paralytic rabies, you can have flaccid paralysis and bitten limb, coma, and death occurs. Then lab diagnosis, what will you do? Antigen detection. Antigen detection is done by direct immunofluorescence. Okay, so gold standard. Best specimen is the hair follicle from the nape of the neck, and viral isolation is done to mouse. Mouse is a suckling mouse. Cell line is a mouse, neuroblastoma cell line, and baby hamster kidney cell line. Antibody detection is done by CSF and the serum. CSF is the most significant one used. What is the antigen which is detected? Glycoprotein G antigen and nucleoproteins. Antibody detection is in RFFIT and FABL. RFFIT and FABL. RFFIT is rapid flow. Sorry. Rapid fluorescence focus inhibition test. RFFIT is rapid fluorescence focus inhibition test. And FABN is fluorescent antibody virus neutralization. That viral RNA detection by RT-PCR. What are the samples? Brain, saliva, and skin biopsy. Negri body detection for postmortem diagnosis and intracytoplasmic inclusion bodies. These are 2 to 10 spherical to oval. Stain for negri bodies, you can use HNE and cellular stain. Okay, celestine can be basic station, methylene blue, methanol. Okay, and you can use man stain also for the granules. Negrivods is basically eosinophilic, and the granules are basophilic. So for granules, you can use Manstin, and for the whole negri body, you can use Celestine or HNE. It's visible by HNE also, right? So no need to use special test. IC will use fluorescent labeled antibodies use. So treatment is no supportive treatment. Okay, only symptomatic treatment is there. Patient is kept in isolation, and all of this hydration is maintained. Okay. So therefore, we come to the end of the discussions. We covered a lot of viruses today. I'm sure you must be forgetting what was HHV3, what was that virus, what was this virus. I know, but do watch this lecture multiple number of times so as to retain what I just discussed. Okay, I'm sure the marathon session will be beneficial for your revision purposes. Like you can get one liners. Now your task is to do MCQs as much as possible if you're preparing for NEET PG, right? And if I'm preparing for the boards, then revise multiple times and think what was there in the lab diagnosis, what was there in the clinical features. You just think multiple number of times, so you get a correct answer, right? So that's it. So today we covered starting from the parvovirus. We covered the parvovirus. We covered the pox viruses. Okay, parvovirus, pox viruses. We covered models from contagious virus. We covered all the herpes viruses. We covered all the herpes viruses. We covered the polio virus. We covered the HPV virus. We covered the rabies virus. Okay, and many other viruses are. This was the virology part one, right? Virology part two. We have other things to cover. Like we have the HIV, the HIV virus. Okay, we have the HIV, we have the viral gastroenteritis also, viral gastroenteritis also. We have other viruses like arboviruses. Okay, arboviruses are a big topic. Okay, and hepatitis viruses, all of this. So let's meet tomorrow in another session on virology, and afterwards, after tomorrow, we'll finish virology. Okay, so that's it, guys. It has been a long lecture, and I'm having a kind of sore throat and throat pain for so long. Okay, that's it. Let's meet again in other lectures. Thank you and good night.